Hello, everybody. Welcome back. If you were with me for paper one, Cram, we are on to paper two. Who is back? Who was here for paper one? Hello, everybody. Hi, Nikki. Hi, Sadia. Hi, Eva. Hi, Sakuna. Hi, Layla. Emo and Sophie. Richard, you're back. Hello. Right, let's just start with some of the questions that I keep seeing in the chat. The first 3 hours are for combined and triple. I'm a little bit concerned that some of the triple scientists are just going to go to the second session. That's not how it's going to work, okay? Hello. If you're triple science, you have to learn everything. You have to learn everything that combined science do plus extra. So potentially you can stay from now until 9 p.m. if you've got the stamina, if you want to cover absolutely everything. Hey, Jessie. But yeah, the first 3 hours are common. So everybody that does combined, everybody that does triple need to be here for the first 3 hours. I've got foundation and higher, but I will flag when it's higher only. So foundation students can just switch off for those questions, okay? But yeah, if your friends were a little bit confused, if your friends are doing triple science and they're just going to come later, you can message them and say, come now, it has started. You need to be here for everything if you are triple science. But things like the brain and the eye, they will be covered in the last hour and a half, okay? Okay, what do I recommend if you don't have the stamina? Remember, this is recorded, so you can watch it later, you can give yourself a break, you can watch it in the morning if you're up early enough. But yeah, everything that we're going to go through is relevant for triple, and the first 3 hours are combined and triple. Okay, you'll notice there's a link in the description. That's the link to the separate stream that's going to start at 7:15. So if you're triple science, you're going to click that link. Okay, but for combined, it's just 3 hours. Okay, and it is AQA, and we will have some breaks. So you've got me for 3 hours, right? So we'll probably do an hour and a half. We'll have a quick little break just to grab a drink, run to the toilet, and then we'll continue for another hour and a half. Yeah, so we will have a break, and then you'll have a little break if you're triple science as well before that starts. Okay, before we begin, this is subscriber only mode. So if you want to use the chat, if you want to get your questions answered and put your answers in for me to see, make sure you are subscribed, like the video for me, turn on your notifications. Can we do a Kahoot? Unfortunately not. We're doing proper exam style questions tonight. We're going to get through as many as possible, okay? Probably ends about 9 p.m. for triple, okay? So if you're triple, you want to be here from now, ideally until 9. If you're combined, it's just 3 hours, okay? Okay, right, should we make a start anyway, guys? Because we've got loads of questions to get through. And then at the very end of my session, I will explain to you triple science students how to get to Joe's session as well. Will we go through everything, says Floof. Obviously, we can't cover absolutely everything, right? Because we have only got 3 hours, but we're going to try and cover as much as we can. We're going to cover a different variety of question types. We're going to cover lots of different content. We're going to focus on things that I think are more likely to come up, like meiosis, like the carbon cycle, things like that. Hopefully it will help you boost your grades. Right, we're going to get going. Use the chat as much as you can. I want to see answers. I don't want to see you just sitting there passively. I want to see you being active. Obviously, if you need me to explain anything in more detail, just let me know. If you've got any questions, just put them in the chat. I will try and get to them. Okay, yes, we are going to have summer school this year as well. Fantastic. Okay, the workbook is in the description to the video, Harry, if you haven't got it already. Thanks, Evelyn. Okay, right, here we've got to name the structures A, B, and C. Now, to do this, look carefully at the diagram so you don't get A and B mixed up. A lot of you are saying sensory neuron. Let me just check. If I trace A back, it does connect to a receptor. So you are correct. If it's going from the receptor, it must be a sensory neuron. And then B is going, if I trace B, it's ending in a muscle. So B is ending in a muscle. So good, it is a motor neuron. So really important we don't get those mixed up. We should know that the sensory neuron goes from the receptor into the central nervous system, and the motor neuron goes from the central nervous system out to the effector. Do you think C is a relay neuron though? It's not pointing to a neuron, is it, C? So C is not a relay neuron. You are correct, there will be a relay neuron. This is the relay neuron here, which is in the central nervous system. But what is C? What part of the central nervous system is that, do we think? I mean, you could have just said it's the central nervous system, but actually specifically it's the spinal cord, isn't it? Yeah, well done. Because the central nervous system consists of the brain and the spinal cord. That's obviously not the brain, that is the spinal cord. Okay, before we look at the other questions, let's just put all this together in a little revision flow diagram. So the way it works, remember, if you do want to revise the reflex arc thoroughly, there is a stimulus, and you can put this down in note form on extra paper, or if you've got space in your workbook. There is a stimulus detected by a receptor, and then we have an electrical impulse. And it's really important we say electrical impulse, isn't it? Not just message, not just information. That's not scientific. And the electrical impulse will go along the sensory neuron, and that will carry it into the CNS. And in the CNS, you have your relay neuron, and that will carry the electrical impulse out along a motor neuron to the effector. Okay. Because we could get like a 4, 5, or 6 marker on this whole thing, right? So the stimulus is detected by a receptor, then an electrical impulse goes down a sensory neuron into the CNS where you have your relay neuron, then it goes out down a motor neuron, and then it goes to an effector. The CNS just stands for central nervous system, and that is your brain and your spinal cord, and relay neurons are found in the CNS, okay? Now there are synapses in between the neurons. So for example, in between the sensory neuron and the relay neuron, there'll be a synapse. Oh, write that in little small for you there. In between the relay neuron and the motor neuron, there'll be another synapse. A synapse is just a gap between two neurons, isn't it? It's just a gap between two neurons. But let's have a look at the next question. How does information pass a synapse or cross a synapse? Because if it's a gap, remember that electrical impulses cannot cross gaps. It's just like physics, isn't it? If you've got a gap in your circuit, the electrical impulse cannot cross. Very good answer from Callum there. Chemicals diffuse, right? And that's all you have to say for 2 marks. You get 1 mark for saying chemicals and another mark for saying they diffuse across. Okay, I can add on from one neuron to the next. Yes, chemicals diffuse, or neurotransmitters. Yeah, if you know the chemicals are called neurotransmitters, you can add that instead. That's absolutely fine. We don't have to use that word though. But it's chemical diffusion. Right, what is the effector in this response? Neurotransmitters is just the name for the chemicals, by the way, that diffuse across the synapse, but you can just say chemicals diffuse across at GCSE. What is the effector? Sadia says 2 easy marks. Yes, let's hope for these easy marks. Right, in this particular response, it's a muscle. And the response will be, if you are asked to give the response, muscles contract. Don't say muscles move. Everybody, that is not good enough. If the effector is a muscle, the muscle will contract and that will cause movement, but we always say the muscle contracts, okay? What is the other type of effector, just so I can check that you know? What is the other type of effector? Not in this example, but just in general. Glands, yeah. So let's just note this down just for revision. The other type of effector is a gland. Now, if the effector is a gland, what is the response? Some of you have told me already. I'll come back to that in a second, Tamina. I've just seen your question. Right, glands. If the effector is a gland, you can say it would secrete a hormone or like a chemical. Yeah? Okay, excellent. Right, Tamina, just going back to the picture, Tamina said, how did we know that A was a sensory neuron? All I did, Tamina, was I kind of traced it back and I could see that A was connected to the receptor. And I know that going from receptor into the CNS is a sensory neuron because sensory neurons carry electrical impulses into the CNS from the receptor. And motor neurons carry electrical impulses out from the CNS to an effector, okay? And yes, MH, is it MH? Yeah. When the electrical impulse reaches the synapse, that's what triggers the release of the chemicals across the synapse, yeah. There could be a 6 marker on this. We've seen it before, which is why I did a quick little summary at the bottom. So we'll just go through it again quickly for those of you that struggle with this. There is a stimulus. It gets detected by a receptor. That will initiate an electrical impulse, which goes down the sensory neuron, then along a relay neuron, then along a motor neuron to the effector, and the effector, which is either a muscle or a gland, will cause the response, okay? And don't forget to mention those synapses, those gaps between the neurons. You might even get a mark for saying chemicals diffuse across the synapses. Okay. I think what is P? P just looks like the kneecap. Looks like, I'm not really sure what P is. Actually, maybe P is a tendon because it looks like it's maybe at the end of that muscle. So maybe P is a tendon connecting the muscle to a bone. That's my best guess. Are you Mank? Yes, I am. Yes, I am. Yeah, relay neurons are in the central nervous system. We also call it the coordinator, don't we, Silverine? The CNS acts as the coordinator. You might have heard that phrase before. Okay, right. Let's move on. Now, I've purposefully left this slide blank because I want you to tell me what this is all about. What system is this? What system is this? Could be, by the way, if you want to see 6 markers on reflex actions, there's absolutely loads on my TikTok that you can watch before your exam tomorrow. What is a synapse? It's a gap between 2 neurons where chemicals diffuse across. Right, this is your endocrine system. So what are all these things that are labelled? I mean, we're going to go through and label them individually, but what are all of them? What are they all called? We'll talk about that now, Mathilde. These are glands, yeah. So these are glands, or to give them their full name, they are endocrine glands. And all of these things secrete hormones. Now, can you tell me in the chat, what is a hormone? Because you might be asked for a definition of a hormone. It's not the digestive system. The digestive system, you'd see like the stomach, the small intestine, the large intestine, and also digestive system is paper 1, right? What is a hormone? It's a chemical messenger. Perfect. That could get you 1 mark. How do they travel? Oh, well done. I've just seen released into the blood. Yeah. They travel via the bloodstream. So that could potentially get you a 2nd mark. And you might even need to say they're released by glands. So let's add that as well. Or secreted just means released. I saw someone ask that before. Secreted just means released. Okay, so hormones are chemical messengers. They travel in the bloodstream. They're released by glands. Right, let's go ahead and label. Now, just to get you guys more involved, I'm going to put some letters on here so you can put your answers into the chat. I'm expecting the chat to go super fast for this because we're going to be good at this, right? Oh, I like that, Mac. I like that. They travel to a target organ. Should we add that as well? Now, while you're putting your answers in the chat, someone asked before, what is the difference between an electrical impulse and a hormone? Well, we've kind of just covered that, right? Hormones are chemical messages and they travel in the blood, whereas an electrical impulse is an electrical impulse and it travels via a neuron or a nerve cell. Okay, so electrical impulses are much faster. Hormones work much more slowly because they have to travel in the blood. Okay, right, I've seen loads of answers. So let's get all of these labeled and let's pray for a question like this tomorrow. So this is the pituitary gland, which is obviously in the brain. Let's go through the rest. I think you've got all the answers in now. This is the thyroid gland in the neck. D is the pancreas. Well done if you got that one right. C is on top of the kidneys. There are two of these, right? These are the adrenal glands. Remember, these are all glands. They're all endocrine glands. They all secrete hormones. Well done, well done. E, yeah, these are the ovaries. So in a female, we have ovaries. And F, these are the testes. So if this was a male, we'd have testes. Could we be asked about food tests? That would have been on paper 1 for AQA, Bob. Okay, now what we're going to do is list some of the hormones secreted by these glands. This is AQA, Justin. Edexcel paper topics are in a little bit of a different order. So, pituitary gland. Let's name at least 2, please. Thyroid gland, you should know 1 hormone. Adrenal glands, 1 hormone. Pancreas, 2 hormones. Ovaries, 2 hormones. And testes, 1 hormone. So let's go through and see if we can do that. Nice. Okay. I'm loving all your engagement, all these answers. Right. We should all know if we're combined, foundation, higher, triple, we should all know the pituitary gland secretes FSH and LH. If you are higher tier, you might also know it secretes TSH, and you might also know it secretes ADH. Okay. But the key ones that everyone should know are FSH and LH. And we've got a question on those ones in a second, okay? TSH, by the way, if you are higher tier, this is thyroxine stimulating hormone, okay? That's thyroxine stimulating hormone, Jazz. It stimulates the thyroid gland to secrete thyroxine. ADH is only for triple science, so triple scientists will do some more questions on that later. Thyroid gland, we should all know that that secretes thyroxine. Well done. Yeah, TSH does stimulate the production of thyroxine. Yeah, but it's higher tier only. ADH is to do with the kidneys and water content of the blood, but that's separate science only. Adrenaline for the adrenal glands. We've got a question on that later. Yeah, ADH is antidiuretic hormone. Water, right? Yeah. Kidneys for triple science. Pancreas, right, we should all know insulin for pancreas and glucagon. And testes, testosterone, which by the way, 1 year they did ask, what is testosterone for? So it stimulates sperm production was the answer that they were expecting. There are other things as well. There are other things like it controls puberty basically in males, but the answer they were looking for testosterone was stimulates sperm production. Ovaries, we've got estrogen and we've got progesterone, which everybody should be able to name because they're in the menstrual cycle. Well done, well done. Okay, don't have to mention the hypothalamus for this one, but if you're triple only, when you do the brain, you'll talk about the hypothalamus later. Do you, Serena? That's really common to get that confused. We're going to do a question on that in a second about glucagon and glycogen, so don't worry. Okay. Yeah, we'll talk about glucagon in a second. Really, for this slide, I just wanted to check, can you label the key glands? Can you give examples of hormones? And do you know what a hormone is? Okay. Excellent. Excellent. Okay. Right, yes, we are going to go over the menstrual cycle. Shall we see what we've got next? Jimmy, that's fine as well. You can say thyroid stimulating hormone or thyroxine stimulating hormone. Either is fine. And thyroxine, that controls your metabolic rate. Well done. Playthings just answered you. Yeah, it controls your metabolic rate. Well done. Okay. So a lot of you were just saying, can we talk about the menstrual cycle? Yeah, menstrual cycle does come up a lot. So we're going to do a couple of questions on that. We've got this first one, which is quite a big 4-marker. Describe the functions of estrogen and progesterone. Now, these are both released by the ovaries. It actually says give the part of the body that releases these hormones. So you can get your mark point 1 just for saying they're released from the ovaries. Okay? But we still have to get 3 marks. So we need at least 3 things that estrogen and progesterone do. I think I'm going to write maybe 4 things because there is more than 3 things. Hopefully this will help Charlotte. Right. Estrogen is from the ovaries. It stimulates LH production. Okay, we could start with that if you want to. I might just go over what LH and FSH do as well, even though it's not for this question. What else does estrogen do? Estrogen, yeah, you could say estrogen causes the uterus lining to thicken. You could also say estrogen inhibits FSH. Inhibits just means blocks, right? It inhibits FSH. Well done to everybody that said that. Zane, Amber, Lux, right. Now, you might think I've already got 4 marks, but the examiner is not going to give me 4 marks, even if I write all of that, because I haven't mentioned anything about progesterone, right? So I have to say at least one thing about progesterone. So what does progesterone do, everybody? Again, it comes from the ovaries. So I've already said it's released from the ovaries. Both of these hormones come from the ovaries. Yes, it does, Alex. Good question. Progesterone, right. Progesterone inhibits both FSH and LH. What else could we say about progesterone? You've got a couple of different options. Progesterone maintains the uterus lining. Excellent. I missed your name then, whoever said that, but I saw your answer. Keeps the uterus lining thick. Remember, we need to maintain the uterus lining in case the egg is fertilized, because if the egg is fertilized, it will need to implant into that uterus lining. Okay, now we've gone through with this question everything that estrogen does and everything that progesterone does. These are all your possible answers. However, I think it might be handy to also just mention, even though it's not relevant to this question, I think we should revise, what does FSH do and what does LH do? Follicle stimulating hormone. Yeah, FSH stands for follicle stimulating hormone, which you don't have to remember, but it does kind of remind you what it does. FSH, yeah, so it causes eggs or follicles, as you can call them, to mature and develop. Yeah, it causes eggs or follicles to mature or develop in the ovaries. And then LH stimulates what? Egg release. What do we call egg release? Ovulation. Good, Izzy. Right, stimulates ovulation. The release of an egg from the ovaries. Well done, well done. Are you allowed short forms for FSH and LH? Yes. They don't expect you to remember follicle stimulating hormone and luteinizing hormone. You can just abbreviate them. Yeah, and ovulation is at day 14. Someone just said that in the chat. Well done. Where do these hormones come from? Where do these hormones come from? Which endocrine gland? Doesn't FSH stimulate estrogen? It does. Do you want to add that as well? We can add that as well in case we have to say 2 things. Right, these ones come from the pituitary gland. Now, the way I remember that is a little bit weird, but I think FSH, LH, PG. Like, I just think of all the abbreviations. LH is an abbreviation, FSH is an abbreviation. Pituitary gland, I could abbreviate to PG, kind of. I wouldn't write that, but so that's kind of how I remember it. They come from the pituitary gland, whereas estrogen and progesterone both have O's, lots of O's in them. They come from the ovaries. Okay. Does LH stimulate progesterone? Yeah, it does. You want to say everything, don't you? This is good. Because all these hormones kind of interact, right? So you've got FSH first, that stimulates estrogen. Then you've got estrogen, that stimulates LH. Then you've got LH, that stimulates progesterone. Yeah. Okay. There you go. I think we've got everything now, Iris. Yeah, we've just added that on. Okay. But do be careful because, like this question, it was only about progesterone and estrogen. So I didn't have to talk about FSH and LH. But hopefully, by doing that little bit of revision at the top, you've got kind of all your hormones now. You don't have to mention the corpus luteum, Sienna. You don't have to use that in your answer. It's basically just, Sienna, if you're interested, the egg cell, if it's not fertilized, it develops into the corpus luteum, and that's where the progesterone comes from. We can just say at GCSE that progesterone comes from the ovaries. You don't have to say corpus luteum. Okay. Okay. Inhibit just means block. It's the opposite of stimulate, if you like. Right. Let's have a look at this next question. Now, this is application because you don't know this, but you're using the graph. So have a look at the graph. Which hormone is likely to stimulate contractions of the uterus when the baby is born? Use information from the graph to give a reason. What do we think? Yeah, we think oxytocin. Why do we think oxytocin? And it does say give information from the graph. Highest during labor. You can't tell that that's labor though from the graph, right? Use information from the graph. Okay, so you could say something like it peaks or rises around week 40 or at the end of pregnancy. So pregnancy is usually 9 months or 40 weeks. And we can see on the graph here that oxytocin, the levels rapidly increase or rise, or it peaks right at the end of pregnancy, 40 weeks of pregnancy. And it is a hormone that stimulates contractions. That's exactly what it is. You don't have to know it, but you should be able to get that from the graph. Okay, this is combined and separate, by the way. Yeah, it goes swiftly up. Yeah. Okay. I feel like I missed a few questions there. If anyone's got a burning question. Yeah, don't worry about oxytocin. No one has heard that before, but you're getting the information from the graph. It's like an application question. Yeah, exactly. This is combined and triple. Okay, some of you were asking me before, why do we want to inhibit FSH and LH? So for example, I think this is an important question, which is why I'm going to pick it out. We said here, estrogen inhibits FSH, and we said here, progesterone inhibits both FSH and LH. Why during the menstrual cycle would we want to do that? Right, exactly. You do want to inhibit FSH so multiple eggs do not mature. You only want one egg to mature, right? And you want to inhibit LH so multiple eggs are not released during ovulation. You don't want multiple eggs to mature. You don't want multiple eggs to be released because that could potentially result in a multiple pregnancy, twins or triplets or quadruplets, which is dangerous for both the mother, the pregnancy is more dangerous, and the babies might not get enough nutrients, etc. So that's why we do want to block FSH to stop any more eggs maturing and block LH to stop any more ovulation. Yeah, you should know that. But to be honest, if you've learned what FSH does and you've learned what LH does, then you can kind of figure out why you would want to block them, right? We aren't going to go through IVF actually tonight. We aren't, but when you have IVF, you do have injections of FSH and LH. Again, if you've learnt the functions of FSH and LH, you already know why. When you have IVF, you need FSH to stimulate eggs to mature, and you need LH to cause ovulation, and then the eggs are extracted and mixed with sperm cells for fertilization. Okay, right. Who is ready to move on? No, progesterone is released even if the woman is not pregnant, Megan. It's released to maintain the uterus lining in case an egg is fertilized. But obviously, if the egg is not fertilized, then the levels of progesterone fall, the uterus lining breaks down, and that's when we have a period. Okay. Can we get your edited version? You mean with my notes on? I don't think that's going to be possible by tomorrow morning, but you've got the video, right? You've got the recording. Okay, let's move on. Let's talk about contraceptives. One example here, suggest one advantage of using a contraceptive implant rather than taking contraceptive pills. Why is FSH inhibited twice? Just to make sure, Dave, make sure that multiple eggs don't develop. You don't have to remember to take it daily. What a good answer. That's the obvious answer. That's the one we're going to go for, isn't it? It lasts longer. It actually lasts, I think an implant lasts about 3 years. So it lasts longer. You don't have to remember to take it. Or you could have said, if you're using the pill, you might forget to take it. Those of you that said it's more reliable, I think you'd have to explain that. I think you'd have to say it's more reliable because if you rely on the pill, you might forget to take it. With the implant, you can't forget because it's implanted in your body. So do explain it. You can't talk about side effects, right? Because actually, the implant and the pill both use hormones. They both use hormones like estrogen and progesterone. So they would both have similar side effects. They both use estrogen to inhibit FSH, and they both use progesterone to inhibit FSH and LH to stop eggs maturing and to stop ovulation. They both have those hormones. So the side effects will be similar, okay? So really all you can go with is you don't have to remember to take it or it lasts longer. Hashim, if you say more effective, I think you need to explain why. You could say more effective because you won't forget to take it. Please be clear with that. Right, can we think of a disadvantage? Why is it a disadvantage potentially to use the implant rather than to take a contraceptive pill? The implant contains hormones and it's just released slowly into your bloodstream. Let's have a look at these answers. Okay, yeah. Painful, someone said. Painful, you could even just say uncomfortable when being inserted, because it does have to be placed under the skin. More invasive. You could go for like there's a risk of infection. Yeah, risk of infection because it is being implanted under the skin. It's more invasive, it's more painful, it's uncomfortable. Yeah, good, good. Don't talk about cost, by the way. The examiners often do not credit anything relating to cost. The other thing is kind of like, you know, if you change your mind, you have to wait longer if you wish to get pregnant. Or you could say like, do you know what I'm trying to get at here? You'd have to have it removed if you wish to get pregnant. Sorry, my writing's getting messy here. I'll try better to keep it neat. Yeah, like it's more of a commitment, but just explain that a little bit further, Alla. So like if because it lasts about 3 years, it does last a long time. So if during that time you decide you want to have a baby, you'd have to get it removed you'd probably still have to wait a little bit after that as well. Or you'd have to wait for like the 3 years. So it's a long-term commitment. And if you change your mind and you want to have a baby, you're going to have to wait quite a long time or get it removed. Gain weight, I mean, so can the pill though, Favour. Allergies, yeah, you could go for allergies, because I do know that on the mark scheme, they allowed things like could cause a rash, like an allergic reaction. Okay. Doctor has to insert, yeah, you have to go to a doctor to get the implant inserted, whereas the pill you can just take yourself. Yeah, that would be okay. Okay, shall we move on? Higher risk of complications. Yeah, just explain it though. All these answers need explaining. Why is there higher risk of complications? That's because it's inserted under the skin. It's painful, it's uncomfortable. There's a risk of infection. It could cause a rash. Just be a little bit more detailed. Okay. Stress. What about stress? Stress because it's painful getting it inserted. Okay, some of you were asking about this. Now, blood glucose regulation comes up a lot on paper 2. So if in terms of predictions, like it comes up a lot for both combined and triple. Explain how blood glucose levels are controlled. Can it be rejected? I don't think so, Lydia. No, can't be rejected. I wouldn't say that. Don't need to know the stages of pregnancy, no. Right, how do we feel about this? So Layla said we're going to talk about insulin and we're going to talk about glucagon. Could be a 6 marker, possibly. So we need to kind of break it up, don't we, into insulin and glucagon. So let me see if you know this. When blood glucose levels are too high, which hormone is released? Good old negative feedback. Yes, it is an example of negative feedback along with temperature regulation, which triple science have to do as well. Good. Lots of us are saying insulin. Right. So when your blood glucose levels are too high, insulin is released. Where from? Where from? And this might be because you've just eaten like loads of carbs, yeah, or you just had a Mars bar, a big glass of fruit juice, a baked potato. From the pancreas, yeah. So I always put that as well. You might not have to if it's a 4 marker, but if it's a 6 marker, I would definitely say the pancreas, okay? So when blood glucose levels are too high, insulin is released from the pancreas. Again, if it was worth 6 marks, I'm just thinking about how you could get your extra marks. Maybe you could even get a mark for saying insulin travels in the blood because it is a hormone, right? It's a chemical messenger. It travels in the blood. And what are the target cells? What are the target cells? Yeah, so I'm just kind of putting extra things in brackets just in case this comes up tomorrow and it's worth like 5 or 6 marks. Where are the target cells? The liver. Yeah, liver and muscles. So liver and muscle cells will take up more glucose. And what do they do with it? Now, I want you to put this into the chat because I want to see if you can actually spell it and give me the right name. Yeah, you do need to know the stages of the menstrual cycle, yes. Well, you need to know the hormones and what the hormones do in the menstrual cycle. Store it as glycogen. T-Tree spelt it correctly. Yes, you've got to spell it correctly. Glycogen. The reason you've got to spell it correctly is glycogen sounds a lot like glucagon. And if you mess your spellings up, the examiner is not going to know if you mean glycogen or glucagon. Okay, so liver and muscle cells store glucose as glycogen. Glycogen is a store of glucose. Okay, now let's do the other half. So when blood glucose levels are too low, which hormone is released? And I do realize there is a diagram. I gave you the diagram just in case people were kind of struggling with this topic and needed extra support. What about starch? Only plant cells store starch. Animals don't store starch. Animals store glycogen instead. Okay. When blood glucose levels are too low, glucagon. Just think of it like this. When the glucose has gone, it's glucagon. Glucagon. Yeah? The glucose is gone. So when blood glucose levels are too low, glucagon is released from the pancreas. And you could say it travels in the blood and it causes the liver to break down glycogen into glucose, and then glucose diffuses back into the blood. So obviously then your blood sugar levels would go back up. Okay, well done. Please get your spellings right, because glycogen, glucagon, very, very similar. Glycogen is a store of glucose in the liver, in the muscles. Glucagon, this is a hormone. It's not a store of glucose. Yeah, perfection. Just the liver. Glucagon acts on the liver only. And it's kind of like muscles are a little bit selfish. Muscles want to keep all their glycogen for themselves. They don't want to give it away. They don't want to let it go back into the blood. So glucagon's target organ is the liver. And in the liver, glycogen is broken into glucose. Glucose diffuses back into the blood. Oh no, you're dyslexic. Oh no, Liv. I would say really try hard with those two words, because if you mix them up, they might think that you don't know which is a hormone and which is a store of glucose. This is negative feedback. Yes. Yes, it is. Definition of homeostasis. Do you guys want that? Homeostasis is maintaining, bit wordy, but I'll write it down. Maintaining a stable internal environment, even when the external environment is changing. And if you need another mark, you could say to maintain optimum conditions for what? Going back to paper 1, homeostasis is maintaining a stable internal environment to maintain optimum conditions for what? Yeah, enzyme action. I mean, enzymes are paper 1, but enzymes are in every single topic of biology, right? Like sometimes students kick off when there's something about enzymes on paper 2, but enzymes are used for every single reaction, right? So, and it's like respiration and photosynthesis can come into paper 2 because they're just such fundamental topics of biology. So don't forget your basic respiration and photosynthesis stuff. Okay. Could you have cells to maintain optimum conditions for enzymes? I think it says enzymes on your spec. Yeah, you could say enzymes and cells, but enzymes is super important. Do you want me to put and cell function as well, so you've got both? There you go. What is negative feedback? Negative feedback is when and you might have to re-watch this if you want to write it down, because again, it's wordy, but if there's a change away from the normal level, it's detected and you have a response to reverse that change. You can use an example in the exam. So for example, if blood glucose rises above normal level, that's detected and it initiates a response to reverse that change. That's negative feedback. Okay, what is glycerol? It's fat, lipids. Lipids contain glycerol and fatty acids. Yeah, there's a response to counteract the change. Good, Iron. Positive feedback is a thing, but it's not used in homeostasis. Positive feedback is where there's a change and then that change gets amplified and increased even further. Not used in homeostasis. Homeostasis uses negative feedback because you always want to reverse the change, right? Bring the level back to normal. Okay, we are going to move on. Yeah, we're doing some ecology towards the end. Why is too high blood glucose a problem? Because water would leave your cells by osmosis, Megan. Water would leave your body cells and go into the blood by osmosis. And then your body cells would like shrivel. Okay. Let's move on. Right. Give the letter and the name of the gland that produces insulin. Now, we've just done this, but I have noticed with these kind of questions, and this could be on a foundation paper, right? Sometimes students ignore the question and they just give the letter or they just give the name. It does say give the letter and the name because they want to know that you can identify it on a diagram. Right, so it is D and it is the pancreas. So do both. Read the question carefully. Give the letter and the name of the gland that produces adrenaline. Let's do adrenaline. Yeah, protein synthesis will be later on with Joe, Susan, because that is triple only. Still on the pancreas. Right, Nadia. Yeah, Nadia's done the next one. So that is C, and that is the adrenal gland, or the adrenal glands, because you've got two of them. Okay. Right, give one effect of adrenaline. Now, adrenaline is higher tier only. So if you are foundation, you don't really have to know what adrenaline does. Just wants one effect, but on the mark scheme, there'd probably be about 10 different things that you could say. So what's like some of the most obvious ones? Okay, increase in blood pressure, someone said. Increase in heart rate. Increase blood flow to muscles. Very good answers. You could have increased breathing rate. You might be asked to give 2 or 3, so it's probably a good idea to write 2 or 3 of these down. Someone said causes your pupils to dilate or your pupils get wider. Increased breathing rate. Oh, did someone just say increase in glucose concentration? Yeah, it does, because it actually causes the breakdown of glycogen, you know, like glucagon does. Has the same effect as glucagon on glycogen. So it does raise your blood glucose levels. I think we've got enough. Yeah, it's a fight or flight hormone. But I wouldn't have said that for my answer, because is that an effect? Not really. Like if you say prepare body for fight or flight, it kind of depends, right? I've seen questions before where it said give 2 effects of adrenaline, and the mark scheme said if the student has said fight or flight, you can give them 1 but no more. Okay, like it might be worth 1 mark, but I would try and give 2 specific effects. Yes, it does prepare the body for fight or flight, but what are the actual effects of that? It's increased heart rate, increased breathing rate. You could even say sweating. Where's my pen gone? You could even say increase in sweating. So just be a little bit careful with that. It might pick you a mark up, but it might want a specific effect. Yeah, sweating is fine. Increase in metabolism. Yeah, Dean, that would be okay. There's so many, isn't there? Dean just said increased rate of reaction, which would be okay because rate of reaction is obviously metabolism. So you could have said increased metabolism. But like, just go for obvious ones. Some of you are giving me some that I'm like, ooh, would it be on the mark scheme? Would it not be on the mark scheme? Obvious things like, you know, right? If you're scared in a fight or flight situation, you know your heart beats faster and you know you start breathing more quickly. So, you know, they're the obvious ones. Like reduced blood flow to the ear, that's random. That's random. We are going to do a question later on quadrats and transects. I've got one on that. What's the hardest topic in paper 2? I think the kidney, but that's triple only. Metabolism is the sum of all chemical reactions. Main one being respiration. It's the sum of all chemical reactions, like in a cell or in an organism. Okay. Yeah, we've got questions on diabetes, actually. You can say increased respiration. Yes, you can. Instead of increased metabolism, you could say increased respiration. Okay. Right. So yeah, someone just said, are we doing diabetes? Have a look at these questions. A doctor thinks that person C has type 2 diabetes. The doctor tests a sample of blood from person C, and the table shows the results of the blood test and the mean results for people who do not have diabetes. Now, if I get a question like this, I go straight to the table and I just check that I understand it. So like this is person C who we think has diabetes. So I'm going to put diabetes type 2 question mark. And this is people who do not have diabetes. And I can ignore cholesterol, but I should be looking at glucose and insulin, right? Because it's a question about diabetes. Give 2 ways the results of the blood test show that person C may have type 2 diabetes. Greater glucose concentration. Well done, Theo. Than, shall I just say normal person? I'm not saying person C is not normal, but you know what I'm saying. Yeah, so they've got a much higher blood glucose concentration, which to me indicates they are not regulating their blood glucose. This could be a sign of diabetes. How do we know, though, that it could be type 2? Insulin levels are also higher. Now, why does that indicate type 2? How do we know from that data that this is type 2 diabetes and not type 1? Yeah, the body cells are not responding to insulin, or they have insulin resistance. Why did I skip cholesterol? It's just not relevant to this question. It's a bit of a red herring. Yeah, remember guys, we're going to do a question on it in a second. But if they had type 1 diabetes, they would not be producing insulin. Now, this person clearly is producing insulin. There's loads of insulin, but their blood glucose is still very high. So this is indicating that their cells are not sensitive, or they're resistant to insulin. Because they are still making it, their pancreas is still making it and secreting it, but their body cells are obviously not responding to it because their glucose levels are sky high. Right, give 2 ways a person can reduce their chances of developing type 2 diabetes. I'm quite keen to see your answers for this one. I want to know if you'll get the mark. I know, Taneem. I do pronounce it funny. Everyone tells me all the time. I try and say it differently, but I can't. I've said it like that for so long. I always put a H in my insulin for some reason. Right, more exercise. You could even give examples of exercise, like go for more runs, go cycling, go swimming. Right, if you just say eat healthy, good diet, healthy diet, balanced diet, you will not get the mark. I'm so glad that so many of you said that. Just so I can highlight it. Balanced diet, healthy diet, good diet, not good enough. Right. Better to say, if you want to get the mark, is reduce or reduced carbohydrate intake. Or you could say like a low carbohydrate diet. Or you could even just say like low sugar diet. Yes, eat less sugar, eat less carbs, because remember, diabetes is about glucose, right? So don't just say healthy diet. If you said lose weight, they only gave you that if you hadn't got any other marks. So that was not the top answer, if you like. You could say lose weight, but they only allowed it if you didn't have either of these. They were really looking for increase your exercise and reduce your carbohydrate intake. Again, just saying lose weight or healthy weight, they only gave you 1 for that if you didn't have either of these. These were the top 2 on the mark scheme. So you could potentially say lose weight. But if you can remember these 2, that is going to be better. You're guaranteeing yourselves the marks. Could you say carbohydrate diet? No. You could say carbohydrate controlled diet. Insulin injections, Fatima, that would be for type 1 diabetics. Type 1 diabetics do not produce insulin, so they need insulin injections. Okay. How does exercise prevent? Because when you exercise, Joy, think about it, respiration. You exercise, you do more respiration to release more energy. So you use up more glucose in respiration. So blood glucose levels fall. Low sugar diet, reduced carb intake. Yeah. Okay. But yeah, if you take away anything, if you remember anything, this comes up tomorrow. Don't just say balanced diet or healthy diet. Low carb diet, low sugar diet. Okay, let's move on. Compare how each type of diabetes is caused and suggest how each type of diabetes can be treated. Now, because this is 4 marks, I kind of can see like you're going to split it into how are they caused and how are they treated. And then you're going to split it into type 1, type 2, type 1, type 2. Just thinking about how I would structure my answer. You don't have to do it like that, but okay. So type 2 can be treated with, we've just said this, so we should all be good. We've already done that, haven't we? But what about the others? Probably having a break at about 5:30, I reckon. Okay, so type 1 is caused by insulin not produced, or you could just say like not enough insulin produced. Okay, the pancreas doesn't produce insulin, or the pancreas doesn't produce enough insulin. What about type 2? How is that caused? Yeah, they weren't looking for you to say it's inherited or it's an autoimmune disease. They just want to know it's because the person is not making insulin for type 1, or they're not making enough insulin. So if you wrote genetic, I don't think you would get the mark. It can be genetic, yes, there are definitely genes linked with it, Floof, but what does that lead to? Well, they don't produce insulin. That's what causes the diabetes. Again, unbalanced. They want you to say cells are not sensitive to insulin or not responsive to insulin. So I can kind of see why you're getting mixed up here. You're kind of saying type 2 can be caused by an unhealthy diet, eating lots of junk food. Well, yeah, that's a risk factor. That increases your risk, but what actually is it? What is the cause of it? It's that your cells or your body cells are not sensitive to insulin. They don't respond to insulin. You can say they are insulin resistant. Yes, that's perfect. Yeah, you can say that. That's absolutely fine. And then type 1, we've got how type 2 is treated already. How is type 1 treated? Yes, we are, Henry. We are going to do that, actually. I added that on at the end. Insulin injections, exactly. Easy peasy. That's easy to remember. Yeah, if your pancreas is not producing insulin, how are you going to treat that? You're going to have to inject insulin. I mean, I could say like regularly before meals, but they didn't need that extra detail. But usually what a type 1 diabetic does is they monitor their blood glucose regularly throughout the day, and they end up giving regular insulin injections. And usually it's before they eat a meal, so that if they do consume carbohydrates like sugars, they've got that insulin prior to eating the meal. Okay. Yes, we are, Emma. Meiosis. I knew everyone would want to do meiosis. Pancreas transplant. Ooh, I wonder if that would be accepted. It's not often done. It's not often done, so they will be looking for insulin injections, but perhaps you could suggest that as a second idea if it's a 2-mark question. But it's usually insulin injections. It's usually before meals that they inject it, by the way, not after meals. Yes, we are going through the carbon cycle. Can you say your body is not responsive? I would be a little bit more specific. It's your cells, right? It's actually like your liver cells and your muscle cells, but at least say cells, not just body. That seems a little bit vague to me. Yeah, well, this number 1, that's what this question is about. So type 1, it's where your pancreas does not produce insulin. Type 2, your pancreas still produces insulin, but your body cells are not responsive. They're not sensitive to that insulin, so your blood glucose levels rise. It would be a pancreas transplant, Archie, because it's the pancreas that makes insulin, not the liver. Why do they inject it before? So the insulin is there, ready to go as soon as they consume those carbohydrates. Otherwise, the blood glucose levels could get too high. Okay. Right, let's move on. Higher tier only. Thyroxine is a hormone produced by the thyroid gland. A decrease in body temperature causes an increase in thyroxine. Explain how the production of thyroxine, so if we increase, spell that wrong. If we increase thyroxine, how could that cause an increase in body temperature? Now, this is really checking that you know what thyroxine does. I don't think I've got any questions on fossils, to be honest. Right. So if we see an increase in thyroxine, that would increase your metabolic rate. Remembering that that's the job of thyroxine. Thyroxine controls your metabolic rate. The more thyroxine in your bloodstream, the higher your metabolic rate. And why would that increase your body temperature? Oh, well done, Silverine. So increase respiration, because respiration is a reaction, right? So that's part of your metabolism. Increase respiration, and respiration releases heat energy, or you could say, as I think Silverine did, is exothermic. Yeah, it's the same thing, Zara. It just means like your base metabolic rate, your normal metabolic rate. You can just say metabolic rate. You don't have to say basal metabolic rate. Remember, metabolic rate is the speed at which chemical reactions occur inside cells. And this is including respiration. So if your metabolic rate increases, the speed that chemical reactions take place at in your cells increases, including the rate of respiration. So these questions are often linked to respiration. This one is because respiration releases energy. We know this from paper 1. Some of that energy is heat. So that would increase your body temperature. Okay. Can you say increases the rate of reactions in the body? Ooh, would they allow that? I'm not sure you'd get the mark, only because you are meant to know, if you're higher tier, specifically that thyroxine, if that increases, it increases your metabolic rate. And that is a key term that they've used on the mark scheme. So I would say increases metabolic rate. And just to add this on, please, for higher tier, because there's not a question on this tonight, but I just want to mention it. The pituitary gland releases TSH, and TSH stimulates the thyroid gland to release thyroxine. So that's how that links together. So thyroxine comes from the thyroid gland, which is in your neck, a little bit lower down. The pituitary gland in your brain releases TSH, which we mentioned earlier. That's thyroxine stimulating hormone, or thyroid stimulating hormone. That travels to your thyroid gland, and TSH stimulates the thyroid gland to release thyroxine, and then that links us back. Thyroxine increases your metabolic rate. What's TRH? You're confusing me there. What's TRH? No, TSH is thyroid or thyroxine stimulating hormone, not adrenaline. Yeah, the pituitary gland is the master gland, Henry, because it releases hormones that affect other glands, right? So like it releases FSH and LH, they affect the ovaries. It releases TSH, that affects the thyroid gland. So it's like the master gland. It's in charge of the other glands. Okay. Oh, TRH from the hypothalamus. Do you have to know that? Is that on AQA? I don't think that's on AQA. ADH, yeah, is antidiuretic hormone. I'm not going to be doing that with you, but Joe, I think, is doing that with you later because that is triple only. Does thyroxine inhibit TSH? Not really. It depends on the level. If levels of thyroxine get too high, then what the pituitary gland will do is stop releasing TSH. So again, you've got that negative feedback. If thyroxine is low, the pituitary gland releases more TSH. If thyroxine is too high, the pituitary gland stops releasing TSH. Okay. No, we won't. Sorry, Mahmood, but that was on paper 1 for AQA. So yeah, if thyroxine levels are too low, Summer, the pituitary gland releases more TSH to make the thyroid gland release more thyroxine. So the more TSH, the more thyroxine is released from the thyroid gland, and the less TSH, the less thyroxine is released from the thyroid gland. No, you don't need to know about the hypothalamus for combined. No, because you don't do temperature regulation on combined. You don't do the kidney on combined, no. Protein synthesis is an example of metabolism. Yes, it's a chemical reaction that happens inside cells. So yes, protein synthesis, respiration, photosynthesis, these are all metabolic reactions. Okay, right, let's look at some questions from module 7. So far, everything we've done, if you're not sure, it's been, oh no, sorry, this is module 6. Everything we've done so far has been module 5. Which is like homeostasis and response. This is now moving into module 6, which is like genetics, inheritance, reproduction, and stuff like that. So this is just a nice table. So we've got sexual versus asexual reproduction. Now, both of them involve cell division. Do they both involve fertilization? Yeah, we've got a question on genetic engineering. Yeah. Protein synthesis could come up for triple. Right, IFA says it's sexual only. Yes, it is. So I'll put a plus there. Because sexual, let's just remind ourselves, it involves the fusion of gametes, yeah, which is fertilization. If you're asked about asexual, you'd say no fusion of gametes or no fertilization. Okay, genes are passed, yeah, and sorry, let's just go back to my notes. I'm thinking of more things to just mention. Involves the fusion of gametes, fertilization, 2 parents. Asexual is 1 parent only. Okay, genes are passed from parent to offspring. Is that in sexual or asexual or both? Sangid says both. Yeah, both. Genes are passed from parent to offspring. Obviously, if it's sexual, then the offspring are getting genes from 2 parents. If it's asexual, they're just getting genes from 1 parent. But both are getting genes from their parents. Offspring are genetically identical to each other. Which one do we think for this? Which one? Only asexual? Yeah. So because all genes or DNA came from just one parent. Okay, so they're genetically identical, or another word for that is clones. Yeah, only asexual. Whereas sexual, they're not going to be genetically identical because you've got DNA from two parents. So you could say they are genetically different. They do quite like a comparison, by the way, of sexual versus asexual. So this is nice little recap. So sexual, let's just go through our key points. It involves the fusion of gametes. It involves fertilization. It requires two parents. The offspring get DNA from two parents, so they're genetically different. Whereas asexual, no fusion of gametes, no fertilization, only uses one parent. And because all the genes or the DNA come from just one parent, it produces genetically identical offspring or clones. Yeah, so asexual uses mitosis. What about sexual? What would we compare that to? If we were doing a comparison and we said asexual is just mitosis or uses mitosis, what would we say would be the comparative point for sexual? Meiosis, yeah, it uses meiosis because meiosis makes gametes. And we're going to do some meiosis revision now. Okay, so it uses meiosis. Which one is faster? Just trying to think of anything else that could come up. Which one is faster and requires less energy? Gametes are sex cells, like sperm and egg. Yeah, asexual is faster. And you can also say requires less energy. Okay, whereas sexual is slower because obviously it's like time to find a mate, time for fertilization to occur, that all takes time. So sexual, we can say, if we're asked for more comparison, we can say it's slower and we can say it requires more energy. Okay, runners are an example of asexual. Yeah, so things like strawberry plants, they grow runners just by mitosis, and then the runner develops like into a new plant. It will be genetically identical to the parent plant because all the DNA just came from one parent plant, but it doesn't require fertilization or fusion of gametes. It's faster and it requires less energy. We're going to talk about diploid and haploid when we move on to meiosis. I've given you some advantages and disadvantages, like advantages of sexual, it's faster and it requires less energy. Advantages of sexual is that the offspring will be genetically different. Remember, genetic variation is always a good thing. It means they're more likely to survive. Like if there's a disease or something, if they're genetically different, it's more likely some of them will be able to survive. Okay, so we've compared our two types of reproduction. Let's do some more questions. We're going to do meiosis in a second. A lot of you are asking me about meiosis in the chat. So chromosomes carry genetic information. Chromosomes are made up of what? Now, again, I've given you a picture just for those of you that are a little bit unsure with this, need a bit of help with this. Chromosomes are made up of what? What are they made from? Yeah, they're made from DNA, deoxyribonucleic acid. They're made of DNA. Now, human body cells contain 46 chromosomes. There are 22 matching pairs, but the final pair does not always match. It is these two that determine the gender or sex of the human. If you are a what, the final pair of chromosomes matches. This is AQA, Faraz, don't worry. Sorry, there's not 20. Why are we talking about 23? Human body cells contain 46 chromosomes, or you could say 23 pairs. Only gametes have 23. Body cells have 46 or 23 pairs. Right, if the final pair of chromosomes matches, you are a female because females have XX. If you are a what, the final pair of chromosomes does not match, that's going to be male, because you should know that the final pair of chromosomes in a male is XY. The final pair of chromosomes in a female is XX. Okay, we all know that. Good. Right, can we draw a labeled diagram to show there is an equal chance of parents producing a baby boy or a baby girl? Now, I can't really get you to do this in the chat, so I'm just going to show you how to do it. So you're crossing a female with a male. Now, we do a Punnett square, right? So the female can produce gametes with either an X or an X, because that's all she has. All egg cells have an X, right? Because females are XX. So gametes will all have X's. Males, on the other hand, produce sperm cells with an X or a Y, okay? So these are my female gametes, my egg cells, and these are my male gametes, my sperm cells. They can be X or Y. And then I have to show all the possible combinations at fertilization. So all I'm doing here is saying if this gamete fuses with this gamete, it would be XX, yeah? But if this gamete fuses with this gamete, it would be XY. And then I could say XX is 50% or a half, and XY is 50% or a half. And obviously these would be girl babies, and these would be boys. So I'm just showing the examiner, it is always 50-50 chance, okay? Both the egg cells have an X chromosome. Remember that male sperm cells can have X or Y, so it's really the sperm cell that determines the sex. Then I show the possible combinations at fertilization, and then I give my final answer where I have to make it clear it's 50% XX, which is female, and 50% XY. It's really important that you set these out clearly because they can be worth a lot of marks, okay? Doesn't matter if female's on the top, no, Harvey. I could have put the XY on the top and the XX on the side. It doesn't matter which way round you put those parents. You can do a percentage, you can do a decimal, you could have even said 0. It doesn't matter because they haven't specified in the question. They've just said equal chance. If they specify percentage, give a percentage. So just check. That would be my advice. Here they just said show its equal chance. So you could give a decimal, a fraction, a percentage. It really doesn't matter. Okay. We are going to do a few more before the break. Right, let's go through meiosis. I know you've all been wanting a bit of help with meiosis. So let's go through and we'll compare meiosis to mitosis, okay? So the first thing that happens, just the same as mitosis, chromosomes are replicated, or we can call that DNA replication, okay? So just like before mitosis, we do have to replicate the DNA or replicate the chromosomes. Now, what you'll notice is that meiosis has two divisions. So division 1 and division 2. Okay, and these are the gametes. Meiosis makes gametes. That's so important. I feel like some students don't even remember that. It makes gametes or sex cells. It does not make normal body cells. It is not used for growth. It only makes sex cells. So like in females, it only happens in the ovaries. In males, it only happens in the testes. It only makes gametes, okay? It's a type of cell division that only makes gametes. Right, now let's do a side-by-side comparison because this comes up a lot. So hit me with your differences. Nicola's just said exactly that. What are the differences? Are plant female gametes ova or egg cells? You can call them either. Ovules, egg cells. The male one is obviously pollen megan implants. Yes, cytokinesis is in both at the end. You have to separate the cytoplasm and the cell membrane, Mr. Sabatron. Right. They both have one parent cell though. Right, meiosis, let's start with something easy. has 2 divisions. Mitosis has 1 division. Okay, and I can see, I've got meiosis on my diagram. I can see it's got 2 divisions, right? Right. Meiosis produces 4 daughter cells. Mitosis produces 2 daughter cells. What else? Genetically identical. Right, meiosis produces genetically different daughter cells. We call them daughter cells. I don't know why. Whereas mitosis produces genetically identical daughter cells. What else? We should still have more to give. Meiosis, haploid versus diploid. Very good. So produces haploid cells, whereas mitosis produces diploid cells. Do we actually know what that means? Well done. Yeah, I've got genetically different. If you're not sure, haploid, in fact, I'll write it down here. Haploid means they have single chromosomes, or half the number of chromosomes. Yeah. I just think haploid begins with a H, half begins with a H. Meiosis is haploid. The daughter cells have half the number of chromosomes, half the mass of DNA. They are haploid. And this is because they only have single chromosomes, whereas in mitosis, they have pairs of chromosomes. So they have double the number of chromosomes in the end. The other one we've not said is meiosis makes gametes, sex cells, whereas mitosis makes body cells. Yeah, so these, you see these 4 daughter cells here? Because this is meiosis in my diagram, these 4 daughter cells are gametes. So this is like 4 eggs or 4 sperms, right? Whereas mitosis makes body cells like skin cells, muscle cells, fat cells, nerve cells, white blood cells, red blood cells. Mitosis makes all your body cells. Meiosis just makes your sex cells, just makes your gametes. Okay? Yeah, body cells or somatic cells. Moon, don't mix up meiosis with sexual versus asexual reproduction. You're mixing things up. Sexual reproduction uses 2 parents. And yes, sexual reproduction does involve meiosis because sexual reproduction needs gametes and gametes are made by meiosis. But you would never say meiosis requires 2 parents, right? That's not true. Like meiosis is happening in your ovaries or in your testes, making your egg or making your sperm cells. Haploid is half the number of chromosomes. Diploid, those cells have pairs of chromosomes. Now, obviously in humans, right? In humans, body cells have 46 and gametes have 23. But the question might not be about humans. It might be a different species. So be careful. If it's about humans, for sure, you can say body cells have 46, gametes have 23. But if it's about onion plants or zebrafish, we don't know their chromosome numbers. So you just need to say that meiosis produces cells with half the number of chromosomes or haploid cells. Leo, you don't really learn about that until A level. It's you can kind of see here, it's like that when the chromosomes line up, they line up randomly on the equator. You don't really need to learn mitosis again for paper 2, but sometimes on paper 2, they have comparisons like this. Meiosis versus mitosis. They do have comparisons like this. Okay, just remember, mitosis is used for growth and repairing tissues. Yeah, whereas meiosis makes gametes. Okay, we're going to try and do a question on, yeah, mitosis happens in all cells. Meiosis is just in like the ovaries and the testes. Mitosis happens like from the minute that zygote forms, it's mitosis all the way. Mitosis to make the embryo, mitosis to make the fetus, mitosis to grow the baby, the toddler, the adult, all mitosis. Meiosis only makes gametes. So I would say learn this table because they do like a comparison. Right. Let's do this question before our break. So the graph shows the mean mass of DNA per cell in arbitrary units, which just means in numbers. Before, during, and after meiosis, between which times is the DNA in the chromosomes being copied or replicated? What do we think? Between which times? So I'm guessing we're looking for 2 times. Between which times? Now, if I'm replicating the DNA, I'm doubling the amount, right? I'm copying it all, I'm replicating it, I'm doubling the amount. So I'm going to be looking, can you see here? Because here I'm going from 4 to 8. I have doubled the amount of DNA. I have replicated the DNA. So it's 3 hours to 4 hours. It said between which times. Well done, well done. Just 3 to 4, not up to 5, just 3 to 4, because you're going from 4 units of DNA to 8 units of DNA. You're replicating the DNA. Which 2 times in the graph show 1 cell dividing into 2 cells? Pain, it doesn't really come up other than to say it's from the testes when you learn about the endocrine system. Okay, so you can see here, hopefully, at this time, you half the amount of DNA because it went from 8 to 4. So if you're halving the amount of DNA, cells must be dividing. But again here, you're halving the amount of DNA because now it's going from 4 to 2. So 6 hours. So you're just applying your knowledge of meiosis, the fact that it has 2 divisions. So cells divide once, then cells divide again. So it would be 5 hours and 6 hours. What is the mean mass of DNA in a sperm cell? A gamete. Remember, a sperm cell is a gamete, it's a sex cell. Now the original cell, the parent cell, started with 4. So it's going to be 2. Yeah, because you've halved it. Just remember, they're haploid cells. They have half the number of chromosomes, half the mass of DNA. And I can see on the graph, it started at 4. It ended down here at 2. What is the mean mass of DNA in each cell in the embryo? So we've just said that the sperm cell had 2 units of DNA. What would the mass of DNA be in a cell in an embryo? Well done. I've not fooled you there, have I? It would be 4 units because remember, you're going to have sperm plus egg. So 2 add 2 equals 4. 4 would be in the zygote, and then the zygote's going to divide to form the embryo. But by that point, we'd be using mitosis. So all the cells would be diploid. They'd have the full number of chromosomes. All body cells have the full number of chromosomes. I can't stress this enough. So in an embryo, these are now body cells. They'd have the full number, the diploid number of chromosomes. So the sperm is haploid, it has 2. The egg is haploid, it has 2. When they fuse together at fertilization, the zygote would have had 4. Then that divides by mitosis. So all the cells in the embryo have 4. Okay. Anything else? A zygote is a fertilized egg cell, Hannah. Zygote just means fertilized egg. You know the first cell when the sperm fuses with the egg? The zygote is that first cell, the fertilized egg cell. And obviously it's really important that the sperm and the egg are haploid because they fuse at fertilization. Yeah? The sperm and the egg fuse at fertilization. and you need to make sure that you get the correct number of chromosomes. So for us humans, the sperm has to have 23, the egg has to have 23, so that when they fuse, we get the correct number of chromosomes, which is 46. Okay? Okay. There's always more application, isn't there, Megan, than we expect. What's happening during 4 to 5? Nothing. Nothing is happening in terms of the DNA. Probably the cytoplasm is increasing in size. Maybe the cell is copying mitochondria, copying ribosomes. But between 4 to 5, nothing's happening to the mass of DNA. But the cell is probably just like preparing to divide, right? You don't have to describe meiosis step by step, by the way, Jossie. You're not expected to have detailed knowledge of the process of meiosis, just that there are 2 divisions and that it makes 4 genetically different haploid gametes. So everything we did on the last slide is what's important. Okay, right, we're going to do this question super quickly and then we're going to have a break just because this is going to take us literally 20 seconds. For now, because we're trying to speed up a little bit, don't have to worry about reading this because it's not really relevant. This is just a maths question. I want everybody to give me an answer in the chat. We've not done any maths yet, really. Calculate the proportion of, these are red blood cells in the image that have an altered shape or a sickle shape. Grab your calculators. You've not had to use it yet. All we need is a proportion. So you're going to be counting the total number, counting the number that have an abnormal shape or a sickle shape. What is the proportion? Are you going to leave it as 4 out of 17 though? Remember, you've always got a calculator. You do it on your calculator, right? So you're going to put it as... Yeah, in maths, you might leave it as 4 out of 17 as a fraction. But yeah, in biology, it's 0.235. That's what I get on my calculator. So I'd round that up probably to about 0.24, or I could leave it at 3 decimal places, or I could times it by 100 and give a percentage. It just says proportion. So I can give it as a decimal. They also allowed percentage. I probably would have just left it as a decimal, to be honest. Don't just leave it as 4 over 17. No, because that's not a calculation, is it? That's literally just you counting. You haven't calculated anything. So don't just leave it as 4 over 17. I just counted the number. So I've got 1 with an abnormal shape, 2 with an abnormal shape, 3 with an abnormal shape, and 4 with an abnormal shape, because that's not the normal shape for a red blood cell. 4 out of, I counted all of them, and I think there were 17 in total. So I just did 4 out of 17. 4 divided by 17. That makes sense. 4 were sickled out of 17. So 4 divided by 17 is 0.24. Okay, guys, we are going to have a quick break. Yes. Can we be back here? I'll write it on the screen at 5:40. I'll write it on, which gives us about 8 minutes to have a quick toilet break and grab a drink. Okay, guys? Yes? No, 10 minutes is enough. I'll see you back here at 5:40. Don't be late. We've got loads more to get through. See you shortly. Hey, everybody. That was a quick little break. Quick little break. The next question we've got is a big genetic cross or a big Punnett square. I'm going to have to shrink my face a little bit so you can actually see the background, I think. So you can see the little key behind me. There we go. Hey, hope you all got a quick drink in, quick snack. Right, this is probably going to be higher tier only because if this was foundation, they tend to give you the Punnett square and all you have to do is fill it in. So it's a little bit easier. This is higher tier because you have to do the Punnett square yourself from scratch. And this is quite a difficult one. So this is really for those students aiming for like grades 7 to 9, right? On the higher tier paper. Right, let's have a look at it and see if we can figure it out. It says persons 8 and 9 are expecting another child. So the first thing I would do is circle 8 and 9. Now 8, if I look at the key, is an unaffected male. So I would probably annotate that on. Okay. And then 9, if I look at the key, is a female. That's why they're in a circle with sickle cell trait. Okay. Now, determine the probability that the child that they have will be a girl with sickle cell trait. You should use the symbols HA for normal hemoglobin and HS for mutated hemoglobin. Let's see if we can work it out. I don't think the answer is going to be 25%, but let's see. So let's start with the male. The male is unaffected, as in normal hemoglobin. So the male is HA, HA. They're unaffected. They don't even have sickle cell trait. They just have 2 normal alleles, and we're told to use HA. Now, the female has sickle cell trait. Now, on the previous slide, which I know you can't see anymore, but on the previous slide, it said that if they have sickle cell trait, they have 1 mutated allele. So 1 normal allele and 1 mutated allele. Now I'm going to get 1 mark just for correctly working out the parental genotypes. Sickle cell trait means they don't have sickle cell. They don't have 2 sickle cell alleles. They just have 1 sickle cell allele and 1 normal allele. They're a carrier, exactly known. They're heterozygous. They have 2 different alleles, or they're a carrier of sickle cell. Right, then I can do my Punnett square. Doesn't matter which way round. I could put the male gametes on the top and then the female gametes on the side. I could have done it the other way with the female gametes on the top and the male gametes on the side. It doesn't matter. Then I'm going to show all the possible combinations at fertilization. Like this. Okay. Now I've already got 3 marks because I got a mark for the parental genotypes. I got a mark for the gametes. So the dad can produce HA or HA, the mum can produce HA or HS. And then I've got another mark for filling in the Punnett square correctly with my offspring genotypes. Right, so what am I actually being asked? Determine the probability that the child will be a girl with sickle cell trait. So these 2 have sickle cell trait. Oh, I think it is going to be 25%, isn't it? So these 2 have sickle cell trait, because they are carriers, they are heterozygous, they have the sickle cell allele. So there's 50% chance of sickle cell trait. But the reason I chose this question was you have to factor in the probability of the child also being a girl. So there's 50% chance or a half chance of having sickle cell trait, and there's a 50% chance or a half chance of being a girl. So I'm basically doing a half times a half, or 0.5 times 0.5. So actually, you guys were right. It is going to be 0.25 or 25%. Yeah, no, that is a sneaky trick. And that's how they boost the question to a higher demand question. It's a grade 7 to 9 question, right? Because you're not just calculating the probability of the offspring having sickle cell trait, you're also factoring in the probability of the child being a girl, or they could say boy, right? If they add that in, you've then got to multiply by 0.5 or 50%. That's the probability of it being one of the two sexes, right? How did you know sickle cell trait was HAHS? It actually said it on the previous slide, Sean. I'll just go back quickly and show you. It said here, can you see? So I was given that information. A person who is heterozygous has both the normal and the abnormal hemoglobin, so they have sickle cell trait. So I knew from reading that to have sickle cell trait, they have to be heterozygous. They have to have one of each alleles, okay? Don't you have to label the Punnett square with person 8 and person 9? You didn't have to, no, to get the marks, but you did have to correctly show what the genotypes were for person 8 and person 9. So this was person 8 and this was person 9. I got a mark for those genotypes. I got a mark for the gametes. I got a mark for the genotype of the offspring. I got a mark for indicating that HAHS is sickle cell trait. And then I got a mark for my final answer, factoring in the probability of being a girl. So my final answer is 0.25. Just before we move on, can you tell me what the definition of an allele is? Because I've been using the word allele. We've been talking about the HA allele, the HS allele, two different alleles. What is an allele? Sickle cell is recessive. Yes, it is. So you have to have 2 copies of the sickle cell allele to actually have sickle cell anemia. So these people don't have sickle cell anemia. They just have what we call sickle cell trait. So these people are carriers. Different versions of a gene. Excellent. Excellent. And I'm guessing we already know that heterozygous is having 2 different alleles and homozygous is having 2 of the same allele. We're all good with that, right? Good, good. We've all got that definition. A mutation is a change in the DNA. A change in the DNA. They asked that actually a few years ago. What is a mutation? It's just a change in the DNA. How do you know that HA is dominant? It actually said on the previous slide, I think. It said on the previous slide. What does the A mean? So H is the gene, hemoglobin, and then the little a is the allele. So the A is the normal allele for the hemoglobin gene, and the S is the sickle allele for the hemoglobin gene. An allele is a section of DNA. No, the definition of an allele is here. I think you're maybe thinking of the definition of a gene. The definition of a gene is a section of DNA that codes for a protein, but there can be different versions of those genes, and the different versions of a gene we call alleles. And remember, these alleles can be dominant or they can be recessive, so you should understand that as well. Lexi, I had to times it because it said the probability of the child being a girl as well. So I had to work out the probability of them having sickle cell trait and the probability of them being female. So I had to times 0.5 by 0.5 because 0.5 was for sickle trait and 0.5 is for being a girl. I had to multiply them together. What is genotype and phenotype? Do we need these as well? So genotype is the alleles you have for a gene. So like HA, that's your genotype. Or big B, little b for eye colour, that's your genotype. It's the alleles that you have for a gene, whereas, where can I fit phenotype? Phenotype is the observable characteristic determined by your genotype and your environment. Did you know that? So genotype is just the alleles that you have for a gene. So we'd represent that with like big B, little b, or HA in this case. It's just the alleles that you have, right, for a gene. Whereas your phenotype is the observable characteristic determined by your genotype and your environment. So like your eye colour, your hair colour, or if you have sickle cell anemia or not. It's the actual observable characteristic. Okay. Genome is just all the genes in a cell, or all the DNA in a cell. No, I wouldn't say phenotype is the one allele you express. No, I wouldn't say it like that. I'd learn this definition that I've written here. I'll just put it in a box. There is, I have got, if you're a little bit struggling with like key terms, there is a video on my TikTok where I do go through all the definitions for like dominant, recessive, heterozygous, homozygous, genotype, like every single inheritance definition. So if you want to watch that, you can watch that after the lesson. Okay. Let's move on using some of these words that we've just kind of covered. The diagrams show one of the experiments performed by a scientist called Mendel. We're on to Gregor Mendel, that famous monk with the peas. In the sentences below, circle the correct term to complete the sentence. So Mendel proposed that flower colour was controlled by inherited factors. The first generation plants show that red is what? Now, if you have a look, we're crossing a red with a white pea plant, but the first generation were all red. This is not triple, this is combined and triple. It's not triple only. So if you cross a red and a white and all the offspring are red, then red is obviously dominant. Good, because it's being expressed in all of those offspring. The second generation plants show that the white factor must be what? Now here, we're crossing together red and red. We've got one white. So what is white? We've got this famous 3-to-1 ratio as well. White must be recessive. I mean, I can show you by putting the genotypes if you want. So this plant is red, this plant is white. And if they're both homozygous, then all of the offspring would have been heterozygous. And then what's happened here is you've got one white plant because this plant must have inherited both recessive alleles. Right, we now call inherited factors what? Recessive, Jessie, means the allele will only be expressed if there's no dominant allele. It's only going to be expressed if there's no dominant allele. Right, so we now call them genes. Obviously, Mendel didn't call them genes because we didn't know about DNA or genes back in his day. So he just called them inherited factors, but we call them genes. This is combined. This is combined. You should still know about dominant, recessive, genes, etc. if you are combined. These factors are passed from generation to generation in what? In gametes. Again, we should know about that if we're combined. This is combined as well. The red flowered sweet pea plants did not all grow to the same height. This was due to what affects the height of a plant. We've just done meiosis. So red is obviously determined by genetics, but what about height? Why are they all growing to different heights? Yeah, environment. E.g., let's say, I don't know, let's think of some abiotic factors, light intensity, amount of water, etc. So that's going to affect the height. So even though they're all red, it doesn't mean they're all going to be the same height because height is also affected by the environment, right? Right, this section is triple only. Why was the importance of Mendel's work not recognized? Why did people, or temperature, competition is a biotic factor, well done, Gnome. Mineral ions in the soil, yeah, exactly. Why did people not recognize Mendel's work? He was only a monk. Love that. You've been studying mark schemes, haven't you? He was only a monk. The reason you've put that in bold is because mark schemes don't accept it. If you just say he was a monk, they mark it wrong. You have to say he was only a monk or he was not a scientist. So people didn't really believe him. Yeah, don't just say he was a monk. The examiner's going to be like, and he was a monk, and you have to say he was only a monk or he was a monk and not a scientist. Don't talk about religion. No, you're mixing that up with Charles Darwin. Right. His work was published in an obscure journal. It did get published, but I think it was in like a gardening journal. Or you could say not published in a scientific journal. He had no aura. The other one that's always on the mark scheme is he only studied pea plants. So some people kind of thought, well, this is only peas. This isn't going to be humans. This isn't going to be animals. He's only studied peas, right? But don't mix this up with Darwin. We're going to do a question on evolution and Darwin in a second. Darwin is when we talk about religion. Okay. How do I know the first flower isn't RR? Because when we crossed them, we only got red in the first generation. We only got red in the first generation. This last bit is just triple, but the rest of it is combined on this slide. Does that answer you, Hazza? If the first red flower had been heterozygous, you would have got some white in the first generation, but all of the first generation were red, showing that that original parent was homozygous red. This is not the end of combined science. Stop saying that. Lack of peer review. You probably could get that, Samir. You could say like other scientists did not support his work. You could say that. Other scientists did not support his work. Where on the spec does it say Mendel is a monk? You definitely are meant to know it, or at least know that he's not a scientist. So yeah, any of these two, he was only a monk, he was not a scientist, his work was not published in a scientific journal, he only studied pea plants, other scientists did not replicate his work, or other scientists did not support his work, that kind of thing. Right, let's move on to Darwin then and see if we can differentiate between how to answer these questions, because this question is very similar to what we've just done on Mendel. And this, by the way, is combined science and triple science. So combined science should still be here. Who proposed the theory of evolution? Who? Chains? That is not true. So stop saying it in the chat or you will get blocked. Right, Charles Darwin. Charles Darwin and someone else. You might be talking about Alfred Russel Wallace. That is triple only. But yeah, there was also Alfred Russel Wallace who did support Charles Darwin. But we do give Charles Darwin credit. We did give Charles Darwin credit. Now, I want to show you for this, I've done something a little bit different. I have literally copied this off your specification, okay? I've copied it from your spec because if you get asked to define evolution, it is one of the most poorly answered questions because students just say like, oh, you know, it's like when organisms change and evolve. Sometimes they stick natural selection. I have copied this off your spec. So I'm going to recommend at this point that you copy this down and try and memorize it. Because sometimes what AQA do is if they put a definition on the spec, they want that exact definition. This is AQA. Yes, it's AQA Sky Blue. If you just say it's a change in a species over time, you might get 1 mark. If you just say it uses natural selection, you might get 1 mark. But if it's a 2 marker, I'd like you all to write this down. I think people that do combined know that this is still combined. So we can stop saying that in the chat. I don't know what you're gaining from saying that. Like everybody has to do evolution and natural selection, right? We know this. What's the definition of natural selection then? Natural selection, it's hard to define. It's kind of, if it's a 1 marker, you could probably get away with saying survival of the fittest, but it's usually worth more marks because you need to usually give a whole story. So you need to talk about mutation, survival, reproduction, and then passing on genes or alleles. So you can't really define it simply. I mean, it's survival of the fittest, but it's the idea that there's a mutation that gives an advantage, and then they survive, reproduce, and pass on that gene or that allele. That's kind of natural selection. But I've got a question on that, I think, in a second, relating to bacteria. Probably not unicorn sisters. You probably would have to say Charles Darwin. Alfred Russel. Yeah, but Charles Darwin got all the credit because he wrote the book. Even though Alfred Russel Wallace did kind of come up with it himself, he was he was writing letters to Charles Darwin, and Charles Darwin was the one that wrote the book, so he got all the credit. Right, many people were opposed to the theory of evolution. Now, don't give the same answers as Mendel. Let's think of some different answers. Yeah, we'll do that briefly, Henry. This is combined and triple. What are we going to say? Religion, right. Went against, we can say religion for this one, because we can't say that for Mendel, because people who are religious are not going to be opposed to breeding experiments in peas, but they are going to be opposed to the theory of evolution, because it does suggest that we evolved from apes, right? So it went against religious beliefs, or you can say it went against the idea that God made all living things. You can also say he had a lack of evidence, but don't say no evidence, because he did have some. He went on a voyage around the Galapagos Islands. He studied lots of different animals like finches. He did lots of drawings. He did collect evidence, but you can say lack of evidence. And you can say, yeah, not enough understanding of the mechanisms of inheritance. Or you can even just say, like, we didn't know about genes or DNA yet, that kind of thing. Yeah, so it's very hard for him to explain. Yeah, DNA hasn't been discovered yet. Exactly, Eloise. It's hard for him to explain because he's kind of saying like, oh, these traits get passed on to the offspring, but he didn't know it was DNA. He didn't know it was genes. We didn't know how inheritance worked yet. So you can go for that route, or you can just say it went against people's religious beliefs. People believed that God made all living things. He didn't have enough evidence. But don't say no evidence. You could probably say didn't know about mutations yet. However, to get the mark, I'd say we didn't know about genes or DNA yet. That's what the mark scheme will say. Yes, we are. We're going to go through both of those practicals. So stay tuned. We're going to do a question on sampling with a quadrant and a transect, and we're going to plan a ruler drop prac. The very last thing we're going to do is the ruler drop prac. Imran, I'm not sure if that's going to be on the mark scheme. These are always the first three answers on the mark scheme. So my advice would be learn at least two of those. Again, Hasta, talk about lack of technology. I would stick with my answers. I always use mark scheme answers. Okay, right. Let's move on. This is a natural selection question, but we've got a maths one first. Many strains of bacteria have developed resistance to antibiotics. Now, you might be thinking, is this not paper 1? But it's natural selection, so it is paper 2. The table shows the number of people infected with a resistant strain of one bacterium in England. Calculate. This means get your calculator out. Don't try and do it in your head. We want a percentage increase in the number of people infected with the resistant strain between 2016 and 2020. How are we going to do it? There's always a percentage question. If it hasn't been on paper 1, there's going to be a percentage change on paper 2, I reckon. So difference over original times 100. So you work out the difference. You divide it over the original, which obviously is from 2016, because that was longer ago. So that's the original. And then you times it by 100. Yeah, change over original times 100. Let me just check if I'm getting the same as you. So 4131 minus 3499. So the change is 632. over the original times 100. Eighteen point one or 18? Yeah? Yeah, brilliant. Right, explain in terms of natural selection why the number of people infected with the resistant strain of bacterium is increasing. You can see in the table, more and more people have this resistant strain. Now, you don't really have to know about bacteria and antibiotic resistance. It's really a question about natural selection. Okay, so why are there more and more bacteria that are resistant? Okay, very good. Abdullah's given me a very thorough answer there. Okay, so we always start with mutation. And remember, they don't choose to mutate. Don't say to the examiner, like, they mutate because people are using antibiotics. Mutations are random. It happens by chance. They are not deciding to mutate because they're at risk or anything like that. Okay, so name the mutation first, Kuki. So there's a mutation, and that must have given rise to antibiotic resistance. So bacteria with the mutation survive and reproduce, passing on the resistance gene or passing on the resistance allele. It is, yeah, bacteria do replicate by binary fission, but you don't have to say that. They're really just testing your knowledge of natural selection. Don't say immune to antibiotics. Bacteria are never immune to antibiotics because they don't have an immune system. They are resistant to antibiotics, which is why they survive, then they reproduce, then they pass on the gene for resistance. Don't just say they pass on the characteristic. Say they pass on the gene, they pass on the allele. You can even say they pass on the mutation. So basically the number of resistant bacteria increases. We don't really have to say that for a mark because I think it said that in the question. It said the number of people infected with the resistant strain is increasing. But yeah, basically, the ones that have this mutation, the ones that have that resistant mutation, they survive, they reproduce, they pass on that resistance gene. So more and more bacteria will have that resistance gene. You could say the total opposite, by the way. You could say the ones without the resistance will die, they will not reproduce, and they will not pass on their genes. But the ones with the resistant gene survive, reproduce, pass on that resistance gene, so more and more bacteria have resistance. Okay, remember that outline for any question on natural selection. There's a mutation. The ones with the advantage survive, reproduce, and pass on that gene. We've done a definition of a gene already, You'll have to go back and find that. We did it a few slides ago. Changes would take time. Wait, I thought changes throughout the lifetime. No, but this Silverine, you're thinking of Lamarck's theory. This is happening when it's passed on to the offspring. Okay, so natural selection is all about the gene is passed on when the species reproduces. Lamarck's theory is triple only. That's different. If you change during your lifetime, it doesn't affect your genes or DNA, so it wouldn't be passed on to your offspring, would it? You definitely cannot say immune, Zanair. You have to say bacteria are resistant. They cannot be immune because they don't have immune systems. They don't have white blood cells. It's not a question about speciation, this one, Bella. It's just natural selection. Good. Mazra. Yes, Sophie. Mazra. Mutation, advantage, survival, reproduction, pass on the allele, Mazra. Yeah, excellent. Okay, let's do a question on genetic engineering. Always a topic that people find hard. I think I'm just going to move myself over here for a second so you can see the whole diagram. The diagram shows how scientists produce genetically modified or GM crop plants. The scientists use a genetically modified bacterium that can invade plant cells. Now we've got a ring of DNA here acting as a vector. What is the scientific name for this ring of DNA? Now, if you look in the diagram, they took a bacterial cell and they got that ring of DNA from the bacterial cell. They're using it as a vector because they're going to put the new gene into the ring of DNA. It's a plasmid. So we're using a plasmid as a vector to carry the new gene into the bacterial cell and then into the plant cell. Well done. At step 1, the ring of DNA was cut open. You can see that here. How do scientists cut open the plasmid? Using enzymes. Yes. Now you can say, if you know the name of those enzymes, they are called restriction enzymes, but you can literally just say using enzymes. That's really all they want. Using enzymes. They cut the ring of DNA using enzymes. They also cut the gene out here using enzymes. That's all you have to say, using enzymes. They are called restriction enzymes. But we're cutting the gene out using enzymes. We're cutting the plasmid open using enzymes. And then if you see here, we're putting, at step 3, we're putting the resistance gene into the ring of DNA. Then we're putting the ring of DNA with the resistance gene into the bacterial cell. Then we're mixing plant cells with the genetically modified bacterial cells, and we're putting them onto agar jelly that contains glyphosate. Why are we doing this? Yeah, you also use enzymes to stick the gene into the plasmid, don't you? Yeah, you've got enzymes that act as scissors to cut the DNA, and you've got more enzymes that act as glue to stick the new gene into the DNA. This is genetic engineering. Right, not quite, Moji. Have a look at what the gene was. We are actually modifying these plasmids and these plant cells. We're giving them a gene for glyphosate resistance. This is combined and triple. So the gene that we have put into the plasmid and the gene that we put into the bacterial cells and the genes that we want the plant cells to take up are glyphosate resistance genes. So why are we going to grow the cells on glyphosate? Exactly, Nish. To check which plant cells have successfully taken up the plasmid or the new gene. Because if they have, they will not be killed by the how do you spell that? I should have said actually at the start, glyphosate is a weed killer. I should have said that because I'm sure you would have been told in the question. So glyphosate is a weed killer, right? And you're genetically modifying these plants to be resistant to a weed killer. Obviously, so the farmer can use the weed killer and it won't kill the plants. But how will you know if the plant cells have successfully taken up the plasmid with the new gene? Well, you grow them on the weed killer, you grow them on the glyphosate, and if they have taken up the resistance gene, they will not be killed, they will survive. If you see that these cells here, if they get killed when you put them on glyphosate, that tells you they have not successfully taken up the plasmid. They have not successfully taken up the gene. Does that make sense? You don't need to know the steps of genetic engineering in detail for AQA, no. you literally just say, cut the plasmid using enzymes, cut the gene out using enzymes, stick the gene into the plasmid using enzymes, and then put the plasmid back into the bacterial cell. It's just all about enzymes. This is a little bit harder because once you've got the plasmid into the bacterial cell, here, you're then trying to get the bacterial cells to be taken up by plant cells. But the key is, I'm trying to make sure you understand this, if the plant cells successfully take up the bacteria, they will be resistant to glyphosate. They will not be killed. If they do not successfully take up the bacteria, they will not be resistant to glyphosate. They will be killed. So it's all about using the diagram, reading it carefully and figuring it out. Glyphosate is a weed killer. So they want the plants to be resistant to glyphosate so they are not killed by weed killer. Yeah, you could say, that's a really good point, Adil. Put the bacterial cells, put the bacterial cells into plant cells at an early stage of development. I mean, these are at an early stage of development, if you have a look, because here, these are just plant cells. Then they're going to divide by mitosis to form adult plants, which means all the cells in the adult plant will have resistance to glyphosate because they'll all be genetically identical cells if they're made by mitosis. Okay. Why would they be resistant? Because Freya, look what the gene was. You genetically modified them with a glyphosate resistance gene. So hopefully they'll be resistant to glyphosate and they'll survive on this plate here when you grow them on glyphosate. Agar is just like a nutrient jelly that we grow bacteria on. It has all the nutrients that bacteria need to grow. Okay. Which scientist invented the classification system shown in the diagram? We've got kingdom, phylum, class, order, family, genus, and species. This is foundation, yeah, and higher. Linnaeus, what was his first name? Can you remember? Oh, we've got a few different, I think is it spelled like that? I feel like I can't spell Linnaeus. Carl Linnaeus. Something like that. Carl Linnaeus. So I remember king, prawn, curry, or fat, greasy sausages. That's how I remember it. King, prawn, curry, or fat, greasy sausages. Carl Linnaeus. Now, who, just to extend you a little bit, who added domain? Who was the scientist that added domain above kingdom? Because he added the 3 domains, didn't he? The eukaryote domain, the archaea domain, and the true bacteria. Who was that? That was Woese, yeah. So Carl Linnaeus did this classification system with 7 different taxa: king, prawn, curry, or fat, greasy sausages. That's how I remember it. But then Carl Woese, it was quite late on. It was in like the 1990s. He came along and he added the domain above kingdom, and he split everything into 3 domains. So eukaryotes, archaea, and true bacteria. Now, obviously, archaea and true bacteria are all prokaryotes. So they have cells without a nucleus. They're like bacteria. And then eukaryotes, it's anything with a nucleus, like plants, animals, fungi. Do you have to know the names of the domains? Yeah, you should know the eukaryote, archaea, and true bacteria. You should also know your kingdoms. So just briefly, you've got the plant kingdom, you've got the animal kingdom, you've got the fungi, you've got the protists, and you've got the prokaryotes as well. So you should know your kingdoms and your domains. Archaeogen, they are prokaryotes, but they're like primitive or ancient bacteria. And most archaea are extremophiles. So they live in really extreme environments. So you might have to say that, so 2 things. They're usually extremophiles and they are primitive or ancient bacteria. Okay, for the archaea. Cloning is triple only. Cloning is not on combined, Precious. So you will be doing that later. Yeah, a lot of you were asking me about triple only. Remember, triple only students have got an extra free 1 hour 45 minutes with Joe at the end, where you will be doing some triple only questions. But remember, all of this is combined and triple. So unfortunately, triple just have to stay for the whole thing. Okay, let's have a look at this. Let's have a look at a kind of evolutionary tree. I'm just going to see if you can do these because I think this is quite common sensey. Which type of present day finch is least closely related to all the others? So we've got all these finches, 1, 2, 3, 4, 5, 6. You've got present day at the top, 1 million years ago at the bottom. They all shared a common ancestor. Which is least closely related? I agree with Riri. Is it Riri that said that? Yeah, vegetarian finch. How do I know that? Well, if I trace it all the way back to 1 million years ago, here's the common ancestor from which they all evolved. The vegetarian finch evolved first on its own. All the rest of the finches shared a common ancestor, which was Q. So all the rest evolved from ancestor Q, which is more recent. The vegetarian finch evolved 1 million years ago from the initial common ancestor. Which branching point, P, Q, R, or S on the diagram above shows the most recent common ancestor of all the tree finches? So read it carefully because these are the tree finches. And I think some students would probably miss that in the exam. There's only 3 tree finches. So which one shows the most recent common ancestor of all the tree finches? So rethink your answer. If you put Q, it is not Q. I agree with Elena, I think. I think it's R, because if you trace all the tree finches back, all 3 of them shared this common ancestor. Whatever R was evolved into the small tree finch, then it evolved into another species, which evolved into the large tree finch and the medium tree finch. So R, it's got to be R. S, those of you saying S is only the common ancestor of the medium and the large. It's not the common ancestor of the small. R is the common ancestor of all 3. Which 2 finches have the most recent common ancestor? There are actually 2 possible answers for this question. How come P is not? If you just have a look at the names in the blue boxes, 3 of them are called tree finches, the others aren't. There are 2 possible answers, I think. Most recent, so we're looking for closest to the present day, where they shared a common ancestor. You could go for medium tree finch and large tree finch because they shared a common ancestor here, which is very recent. But you could also go for the mangrove finch and the woodpecker finch, couldn't you? So 2 possible answers using that diagram. Yeah, because if you look at P, they shared a common ancestor at P. And again, that was about the same length of time ago from the present day. So there's actually 2 possible answers. I think there was only meant to be 1 answer. I think my diagram's a little bit wrong. But on my diagram, there are 2 possible answers because S and P are at the same level. So the mangrove and the woodpecker shared a common ancestor. The medium tree and the large tree finch shared a common ancestor about the same amount of time ago. Okay. Describe how you would use a 0.5 by 0.5 meter quadrat frame and a 30 meter tape measure to obtain results on the number of different plant species growing from one side of a marsh. A few of you were asking me to do this. This is required prac. This is sampling. But I know this is going to be transect. How do I know? Well, they are not asking me to find a population size. They are asking me to find the number of different plant species growing from one side of a marsh to another. This is definitely combined and triple. Yes, exactly, Alex. If I've just got a field and I just want to know the population in the field, I'd just use a quadrat, right? I'd just use random sampling with a quadrat, lots of quadrats, count the number of different species. But this says I actually want to know the number going from one side of a marsh to another. I'm going from one place to another. I'm obviously trying to see how the marsh affects. So I have to go from one place to another. That's a transect. Yes. So it's not going to be random sampling as such. It's going to be placing my quadrat along a transect at regular intervals. Yes. Now, what is a transect? I've got a mark scheme to show you for this one. A marsh is like a boggy, waterlogged area. What is a transect? Like literally, what is it? If I said go to that marsh and lay a transect, what would you do? It's probably longer than a meter ruler. It's probably a tape measure. Yeah, it's like basically a tape measure. You could even just put a rope down. Straight line tape measure. Yeah. Right, let me show you the kind of answers they were expecting. Right. They want you to realize with this question that you're going to have to produce a transect. So you could literally say, lay a tape measure across the marsh. 2 marks already. So use of a tape measure to produce a transect. You want to go from one side of the marsh to the other. So you're going to have to place your transect across the marsh in a straight line. So let's imagine, here's my marsh. I want to go from one side to the other. So I might lay my transect there. Then I'm going to use my quadrat, but I'm not just going to do it randomly. I'm going to be placing it along my transect. So I could, oh no, yeah, you could do it there, 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 yeah, etc. From one side of the marsh to the other. So place quadrats along the transect, and then you're going to count or record the number of different species, because it says, I want to know the number of different plant species. So use the information in the question. You're going to be placing your quadrat down and basically saying how many different plant species in that quadrat, count them, then place it again further along. How many different plant species in that quadrat, count them, record them. Place the quadrat at regular intervals. I don't know, e.g. every 2 meters. You don't really want to do it randomly because you might get, if you do it by random, by chance, you might get all your quadrats on one side of the marsh. And you want to make sure you've got quadrats all the way along from one side of the marsh to the other. So do read the question carefully. The clue is it's a transect. You're going from one place to another. A large number of quadrats, always, whether it's random sampling or using a transect, always use a large number of quadrats. If you're going to give a number, at least 10 quadrats is what I would recommend. Repeat the transect several times. So look, you could do it again here. You could do it again here. At least 3 times. Now, in terms of placing your transect, they will let you place your transect at random places across the marsh, or you could even place your transects like, I don't know, 5 meters apart from each other. So you could say place your transects at random, or you could even have like a set distance between your transects. But the key things to remember, you need lots of quadrats. Look out for when it's not random. This is a transect. You want regular intervals across the marsh. So lay your transect, place your quadrats, regular intervals, large number of quadrats, repeat your transect. I mean, I don't think this is a particularly hard practical, but sometimes you need to think, am I just random sampling? And that would be like if you've just got a field and you just want to know the number, you just want to know the population, or is it a transect if you're going from one place to another? But with both, you still have to use quadrats and you still have to use a large number of quadrats. Yeah. What if it isn't a transect, Maisie? Then that's when you go down your route of saying use a random number generator to generate coordinates, and then place your quadrats at the random coordinates. But again, you'd have to say use a large number of quadrats or take many samples. But with that kind of question, it's really important that you say place your quadrats at random. So use a random number generator to decide where you're going to place your quadrats. Okay. Could you not just use one quadrat? Not really, because how are you going to get information across the marsh if you just place it once? You're going to have to keep placing it down, going from one side to the other. Yeah, this is ecology. Don't throw the quadrat, no. You're either going to place it at regular intervals along a transect, or you're going to use a random number generator to generate random coordinates and put your quadrat at those random coordinates. Remembering to do at least 10 minimum. Do you need to calculate a mean? You didn't have to say it for this question. That tends to be more. Who asked that question? That tends to be if you want to find the population size. If you want to find the population size, then you'd have to work out the mean number per quadrat and then multiply it by the area of the field. Because you'd have to know the mean, wouldn't you, per one quadrat, work out the mean per meter squared, and then you could multiply it by the area of the field. But that's if you're trying to find the population of something in a field. Okay. Let's move on. Right, so let's fill in the table, please, guys. I'm going to number them. This is ecology, by the way, if you've not noticed. So we've got a food chain: grass, snowshoe hare, lynx. All we've got to do is fill in the table. So the examiner is just checking that we understand the words: primary consumer, producer, herbivore, etc. We're going to do one at the end where we have to do IV, DV, and CV. Yeah, we're going to do that kind of practical at the end. Right, so the primary consumer is the snowshoe hare. Come on, guys, let's remain focused, get our answers in. This should not be hard. Try and say snowshoe hare, not rabbit, Zane. You probably wouldn't be penalised, but it is a snowshoe hare, not a rabbit. The producer is the grass, because it's photosynthesizing, it's producing its own food in the form of glucose. The herbivore, again, try and say snowshoe hare. Otherwise, the examiner might be like, why are they saying rabbit? What are they talking about? The carnivore is the lynx. And you can repeat your answers more than once. Some students would make random answers up because they feel like they can't put the same answer twice. What's glucose? I hope you're joking, but it's a sugar. It's a sugar made in photosynthesis. The prey, again, is the hare, and the predator that hunts and kills, that's the definition of a predator, hunts and kills, that is the lynx. Okay, when the snowshoe hare eats the grass, almost 90% of the biomass of the grass is lost. Give 2 ways biomass is lost. Now this is, I think, a triple only question actually, but the combined people should still stay here because the rest of it is combined. What were we going to say? Right, you could say lost in respiration or lost as CO2. Waste is a bit vague. You can split waste down. You can say lost in feces. Waste would only get you 1 mark. Excretion would only get you 1 mark. You can split it down. Yeah, you could say not all of the grass is eaten, e.g. maybe the roots. So maybe the hares don't eat all the grass, maybe they don't eat the roots, so obviously that biomass is wasted. The snowshoe hare will lose some biomass in its urine or urea, it will lose some biomass in its feces, or because not all grass is digested, and if it's not digested, it's just going to pass straight through in its feces, and you can say lost in respiration. Yeah, you could say digestion for this one. But I wouldn't just say waste. They're probably going to limit you to 1 mark if you just say waste. So try and learn separate answers. Try and say lost in feces and lost in urine, because that's 2 separate marks. Yeah, feces, not all eaten. Biomass is just the mass of living material. So the grass contains biomass, but not all of that biomass gets transferred to new biomass in the rabbit or the hare because of these reasons. Digestion is just getting rid of feces. Yeah. So not all the grass can be digested and it passes straight through in feces. That's digestion. Movement wasn't on the mark scheme, but movement is, I suppose, the same mark point as respiration, because for movement, they require energy. To get that energy, they have to respire. So movement is really lost in respiration. Producer is a green plant that photosynthesizes and produces its own food in the form of glucose. Okay, right, we need to get onto this. Let's do the water cycle first, because that's super easy. Let me label them all. Let's do the water cycle first. Cycles haven't come up for a while, so could come up this year. X, Y, Z. Let's go for W and V. Had to think what was before W in the alphabet then. Right, let's do the water cycle first. All I want you to do is literally label the processes. This is really easy, water cycle. So put the letter in so I know, like Lara has. So X is evaporation. You've got liquid water evaporating into water vapor. Yeah, I think the carbon cycle is more likely to come up than the water cycle. Z is precipitation, which could be rainfall, but it could also be snow or sleet or hail. W is transpiration, which is the evaporation of water from leaves or from plants. Y is condensation. Yeah, so that water vapor condenses back into liquid water, which forms clouds. Yeah, what are we calling V? I always call it percolation, but they do allow like runoff. Yeah, runoff or percolation. Runoff, yeah, percolation, good. Yeah, through flow, yeah, I think they'd allow through flow. I'm not sure about drainage. I'm not sure about that one. I always say runoff or percolation, that it kind of trickles through the soil and through the rocks and makes its way into bodies of water. Let's do the same for our carbon cycle. So I'm going to put some letters on this. This is more complicated. I'm going to put that one as A. I'm also going to put that one as A. And I'm also going to put that one as A. And then I'm going to put both of these as D. Percolation sounds more scientific. Yeah, always go for the more scientific one. Oh, aqua. Joe will be doing some questions on the I with you later, so make sure you ask him your I questions. Yeah, so A is respiration. Yeah, because look, green plants, and this is so easy to get marks on. You can pick up marks, honestly. It's not as hard as you think. Green plants respire. That releases CO2. Animals also respire. That releases CO2. Microorganisms also respire. That releases CO2. Easy. And then B is photosynthesis. And that's only going to the plants, right? Plants photosynthesis or photosynthesize, and that absorbs CO2. Yeah, you can say it in any order because it's a cycle, so it doesn't matter what order you give your answer in. What's D then? Decomposition or death and decomposition or decay. Yeah. Now that's obviously done by microorganisms. How does that release CO2? Well, as microorganisms cause decay, they respire, which releases CO2. What about C? Have we got C? Consumption, yeah, or I was just going to say feeding. Now, you might have to word that out, okay? We've done it really simply, but it's so easy to word out because you know these processes, you know respiration and photosynthesis from paper 1, yeah? So green plants respire, which releases CO2. Animals and microorganisms respire, which releases CO2. That CO2 goes into the atmosphere. Then you say plants absorb CO2 for photosynthesis, and then animals eat the plants. And then all you've got to add on is animals and plants die and are decomposed by microorganisms, which obviously takes us back to they respire, which releases CO2. Yeah, the only thing missing off here, which we can add on, is if the green plants and the animals don't decay, they may, if they don't decay and they're subjected to high heat and pressure, they may form fossil fuels. So what would this label be? What would E be? Some of you are asking me about that one. No, microorganisms don't take in CO2, they release it. So they eat no, they don't eat. They decay the plants and animals when they're dead and take in the carbon compounds and then respire, which releases CO2. Yeah, so E would be burning or combustion. Exactly. So you could add that one on as well. So use that little checklist. Just think, have I spoken about respiration, photosynthesis, feeding, death and decay, burning, or composition? Yeah, carbon cycle's a bit of a prediction. I think that could come up. So you can always go back to this little slide and write down a few more notes if you want to. But that's your little like checklist. You should have spoken about A, B, C, D, and E, but just obviously link them together. Are they releasing CO2 or absorbing CO2? Let's have a look at some questions. So give the letters of 2 arrows in the diagram which show respiration. Now there are actually 3 possibles that you can give, but you only have to give 2. If you don't understand how to word it in the exam, I would suggest if you look on my TikTok, it's all in playlists. So in my module 7 playlist, in my ecology playlist, I've done like a written answer for the carbon cycle. So C, yeah, C is animals respiring, releasing CO2. Yeah, D is plants respiring, releasing CO2. You could have also gone for B because that's obviously microorganisms respiring. Give the letter that shows decay. Now this is a bit different to my diagram. You've got death labelled, but which one would you say shows decay? Floof says B. Yeah, it is B. So B is decay, but it could also be respiration because as microorganisms decay, they respire, which releases CO2. So you'd say something like microorganisms decay dead plants and animals or decompose dead plants and animals. And as they do so, they release CO2 during respiration. Okay. A is obviously combustion or burning. Okay. Yeah, plants decay, microorganisms decay plants, unless they're buried in layers of sediment, like it's happening here to form the fossil fuels. And what would F be? Would it be sedimentation or something? What do you learn in chemistry? It's like layers of sediment build up, don't they? To form fossil fuels, like layers of sediment build up on top. Is that sedimentation? They didn't ask you what F was, by the way, in the exam, but I'm going to say like maybe sedimentation. Because I think layers of sediment build up and then you get high heat and pressure and then it forms fossil fuels. So the lines are all showing processes, labelled processes. Why is there an arrow from plants to CO2 in the atmosphere? Oh, it's going the wrong way. This one. D is photosynthesis. It's kind of labelling the wrong thing. Plants going to CO2 in the atmosphere is photosynthesis, isn't it? No. Sorry, said that wrong. Plants going to CO2 in the atmosphere is respiration. So D is respiration. You can tell I've been teaching for too long tonight. This one is photosynthesis. So CO2 going from the atmosphere into the plants is photosynthesis, because plants absorb CO2 in photosynthesis. The only way to remove CO2 from the atmosphere is photosynthesis. Plants absorb CO2 for photosynthesis, but they also release it in respiration. That's why there's two arrows going from the plants. Is it fossilization? Is that what you call it in chemistry? Fossilization. I mean, you weren't asked. I don't think you need to know that for biology, really. Okay, the concentration of CO2 in the atmosphere has increased over the last 150 years. Give two human activities that increase the concentration of CO2. Could what be digestion? E could be like feeding and digestion. E could be on there when animals eat plants. Right, Alex knows. Who are we going to go for? The obvious, the two they always want. Burning fossil fuels like coal, oil and gas releases CO2. And deforestation, cutting down trees on a large scale. They're the two they always want. Oh, I wonder, yeah, you could have destroying peat bogs. That releases CO2. Good shout. Good shout. Got to allow that. Cattle farming. Yeah. Increase in cattle farming. You could have that, because the more cows, the more respiration, the more CO2. So if we're grazing more cattle, you've got more animals respiring on the planet. Respiration does release CO2. Yeah, why not? But I would say an increase in cattle farming. But the top two are going to be burning fossil fuels and deforestation. Rice farming, no, Parry. Rice releases methane. We're going to talk about that now. So when we grow more rice in paddy fields, that releases methane, not CO2. Driving would be fine. Any example of using a fossil fuel would be fine. So driving cars, for example, you're burning fuel. That's a fossil fuel that releases CO2. Draining peat bogs, yeah. Destroying peat bogs, yeah. Right, let's just talk about global warming and greenhouse gases. What are the top two greenhouse gases? Name them for me. There are more than two, but what two do you have to know for GCSE biology? Peat bogs is just where plant material has not decayed because it's waterlogged. So it's undecayed plant material. Yeah, top two for biology, I would say, are methane and CO2. You might also know from chemistry, nitrous oxides, even water vapor, you might know. But for sure, for biology, we should know these two. Now, CO2, as we've just said, comes from burning fossil fuels, and it's also increasing due to deforestation and destroying peat bogs and grazing cattle. Methane, growing rice in rice. Have you heard of paddy fields? Do you guys call them paddy fields? And also from grazing cattle. Yeah, I think it's belching, not farting. But basically cows, I'll just explain it if you're not sure. Cows have bacteria that live in their digestive system. And these bacteria produce methane during digestion. And when a cow, I'm sure it's burping, not farting. When a cow belches or burps, methane is released. So methane, the two sources of methane, grazing cattle, because it's produced in their digestion, and growing rice. Where we grow rice in paddy fields, that releases methane. So you've got your cows and your rice for your methane, and then you've got your burning fossil fuels and deforestation for your CO2, okay? They're the ones we should know. Grazing just means, when they're eating the grass, like they're out and about on the field eating the grass. Now, we're just going to quickly go over this, although I have just posted this if you want to watch the vid on YouTube. But basically what's going on is the sun's rays, just follow this with me, you've got the notes. The sun's rays enter the Earth's atmosphere. So you've got light energy and heat energy from the sun. Some of the heat is reflected from the Earth's surface and some of the heat escapes into space. But Some of the heat gets absorbed by the greenhouse gases in the atmosphere, or you could say trapped by the greenhouse gases. And then what happens to it? What happens to it? What's the problem? It's getting trapped by these greenhouse gases, the CO2 and the methane in the atmosphere. It's getting trapped, and then what's the black arrow showing? Yes, the heat energy, I think it's best to say re-radiated. Re-radiated. Has anyone said that before? Because it's kind of radiated off the Earth's surface. Some of it escapes into space. Some of it gets trapped by CO2, methane, nitrous oxide, and then it gets re-radiated, re-emitted. Is that what you say in physics? I think in bio we tend to say re-radiated, but I'm sure they'd accept physicsy answers, right? Re-radiated back to Earth, and this causes global warming, which is the idea that the average temperature of the Earth is gradually increasing because more greenhouse gases means more heat energy gets trapped and more heat energy gets re-radiated back to Earth. Re-emitted, we're all saying re-emitted. We use re-radiated, re-emitted, I'm sure is fine. Yes, you do need to know a bit about eutrophication. You do. I don't think we've got that in our lesson tonight though, unless Joe has it. You think re-emitted sounds better. I'm sure you can use re-emitted if that's what you say in chem. But yeah, just to say, learn your greenhouse gases, know your sources of them. Where does the CO2 come from? Where does the methane come from? And understand that the problem is with more greenhouse gases, more heat energy is getting trapped and then re-radiated back to Earth. Earth's temperature is increasing. This is all suitable for single science and combined science. Right, let's do a few questions. Tropical rainforests are being cut down to provide hardwood for furniture, to make way for roads, for agriculture. So, e.g., growing crops or grazing cattle. You might have to give reasons for deforestation. So you've got a few there. Wood to build furniture, to build new roads, for agriculture, to grow crops, to graze cattle. You could even say to grow rice. Now, in the 1980s, they were being destroyed at a rate of 15 hectares per minute. Calculate the number of hectares destroyed in a day. Wow, you are so fast. So it's 15 per minute. So I'm going to times it by 60 to get it per hour. So 900 per hour. And then I'm going to times that by 24 because there are 24 hours in a day. Yeah, exactly. I agree with you guys. You were all faster than me. I was too busy drinking coffee. Yeah. So it's just testing, you know. There are 60 minutes in an hour and then there are 24 hours in a day. The gas carbon dioxide can contribute to the greenhouse effect. Explain how deforestation can contribute to the greenhouse effect. 3 marks, quite a long one. Right. Good start there, Umar. So fewer trees means less. A lot of people forget this one, actually. A lot of people forget that if you cut down trees, there's fewer trees, therefore less photosynthesis, less CO2 is being absorbed, decrease in CO2 intake. Yeah. What else are we going to say then? Because we've only got 1 mark. We need some other reasons though, guys. You are right, more CO2 would then be in the atmosphere and CO2 is a greenhouse gas. But how else can cutting down trees? Yeah, Nwando, I'm waiting for those answers. Okay, we're all just talking about. But also, don't forget, guys, Once you've cut down those trees, the trees are often burnt, which releases CO2, or the trees are decayed by microorganisms, which respire and release CO2. And so then you've got your increase in CO2 in the atmosphere. So there are kind of three things to talk about with deforestation. Why does deforestation increase CO2? Well, you've got less trees, less photosynthesis. So yes, you all said less CO2 is being absorbed, but also once you've cut down the trees, they might be burnt, which will release the CO2 that they were storing, or they might decay by microorganisms. That releases CO2 because microorganisms respire. Then you're going to get an increase in CO2 in the atmosphere. And you are all right. Obviously, CO2 is a greenhouse gas. We've just said it traps the heat energy and re-radiates it back to Earth. But we needed three things linked to deforestation. Oh, I like that one. The machinery being used might actually release CO2 as well. Okay. Can you say the area might be... Oh, possibly. Yeah, that's actually a really good point. Yeah, I think you could. After you've cut down those trees, you might use that land to grow rice. You might use that land to graze cattle. And that could contribute to global warming because grazing cattle and growing rice releases methane, which is also a greenhouse gas. I love that answer. Well done. I love that answer. Yeah, if you're going to use the land to grow rice or graze cattle, increase in methane. Even grazing cattle, increase in CO2 because cows and bulls respire, which releases CO2. I love that. Trees store carbon. When cut, the trees release. Yeah, but you have to say how they release the trapped carbon. It's because they decay, Miri, or they're burnt. That's how the trapped carbon gets released. Yeah, I think you probably could get credit for the machinery because the machinery used to cut down the trees obviously will use some kind of fuel like oil. And yeah, that would release CO2 when burning that fuel. Yeah, but these would be the top three answers is what I'm saying. One result of the increased greenhouse effect is global warming. Describe two possible effects of global warming on the world. Now, there's a big list of answers here. I wouldn't say increased temperature because they've already told you in the question global warming, which is pretty obviously increased temperature. So, rising sea levels, melting ice caps. They might be the same two marks because they are very similar. Flooding, droughts. You could even just say extreme weather events, more extreme weather events. Yeah, more fires, more droughts. You could say loss of habitats. Because obviously if it's a low-lying habitat, it could get flooded when the sea levels rise. You could say reduction of biodiversity because a lot of plants and animals might become extinct. The other one you can always have is change in migration patterns, like birds migrating at different times to what they usually would in the year because the temperatures change, changing rainfall patterns, very good, extreme weather events, shrinking glaciers, more forest fires, global dimming. Is that something you do in chemistry? We never really mentioned that in bio, but it would be accepted. Change in migration, say Nwando, change in migration patterns, more wildfires. Yeah, brilliant. Excellent. Okay, last question. So we're not running too much over, don't worry. Now, this is the kind of question where I would use my little trick. So if you've had me before, maybe if you came to Cram Paper 1, whenever I get asked to plan a practical like this, I always think IV, DV, CV, repeats. That's my hack. So a student investigated whether using the right hand or the left hand had an effect on reaction time. So IV, what am I changing? This is how I plan. Right hand versus left hand. Okay. DV, what am I measuring? Reaction time. And then I'd probably think, How? How am I going to measure reaction time? Well, what have I learned? Ruler drop test. Probably going to use the ruler drop test, yeah? Then I'm going to think CVs. What do I need to control? So then I need to think anything else that could affect reaction time, I need to keep the same. So same age participants. You could even say same gender in case that affects reaction time. Same amount of sleep. I don't know. Same person. I don't know what you mean by same person. You're going to do what repeats. So you're going to be using lots of different people. Same age, same gender, same height, ruler is dropped. That's a really good one. Because you can't drop it from like way up and expect them to catch it and then drop it from really close. That's a really good one. And then I always think of repeats. So you could think of like use a large number of people. Like if you're using people like we would be here, the more people, the better. You could even think about 3 repeats with each person. That would make it really good. That's going to push me up into the highest bracket because I'm going to use a large number of people. The more people, the better. And maybe I'll get each person to do it 3 times with their right hand and 3 times with their left hand. Same noise levels, yeah, same level of disruption. That's a really good one. Temperature, I'm not sure temperature would affect reaction time. Right, once I've done that, then I can do my answer. And to be honest, I've probably already picked up quite a few marks. I might have actually got 4 marks just from doing that plan. Because I've said I'm going to do the ruler drop test, which is a basic method. I've said I'm going to do it with people's right hand and left hand. And I've got like 3 or 4 control variables. I actually might get 4 marks. So always think, what is my IV? What am I going to change? What is my DV? How am I going to measure it? What will I have to control? And then just the more repeats, the better. So large sample size, large group of people, make them all repeat it. Same length ruler. Yeah, always use a 30 centimeter ruler. Yeah, you could say that. Use the same hand each time. Well, you are changing the hand. That is your IV. You're going to get them to do it in their left hand and their right hand. So you might do like 3 repeats with the left hand and 3 repeats with the right hand. Okay, let's have a look. Now, this is an extended response question. It's not that you have to have every single bullet point, okay? But you have to have a method that the examiner thinks would work. And usually to get the highest marks, it's all about control variables. So you need to use a large number of people. Well, we actually already had that over here. Do a reaction time test. Again, repeats, really important. Use their right hand first and then repeat it with their left hand. Well, we already had that over here. Method of studying reaction time. Now they're going to expect you to do a ruler drop test. Basically, one person drops the ruler. The other person sits with their arm on the edge of the desk and catches it. There's really not a lot to explain. They really don't care about the detail there because it's so basic. It's a ruler drop test. One person drops the ruler, the other person catches it. That's like your whole method, right? They really don't care about that. The important things are you've got a large group of people. You are repeating it. You are doing it with both their right hand and left hand. So the independent variable is studied. You record the distance the ruler was caught. You can then look up the reaction time. They will give you extra credit for calculate a mean reaction time because you're going to have a lot of data. And then same age, same gender, drop the ruler from the same height, even same time of day so you're not tired at the end of the day. Control how much sleep they've had, control how much coffee they've drank because that could affect reaction time. You could say control the noise levels or control distractions. The student only tested right-handed people, I suppose, because they're trying to see, will they have a faster reaction time with their right hand, because that's their dominant hand, as opposed to their left hand, because that's their non-dominant hand. So it just makes it a bit easier. Yeah, Nwanda, it makes it easier. If you test left and right-handed people, that would complicate it. Because if they're left-handed by nature, they'd be faster with their left hand. If they're right-handed by nature, they'd be faster with their right hand. Now, if you only test right-handed people, you would expect all of them to be faster with their right hand and all of them to be slower with their left hand. So it just makes it easier to analyze the results, but you still have to get them to catch it with their right hand and catch it with their left hand, because your independent variable is the effect of hands. So you need to do both so you can compare the reaction time. You could say, yeah, you get extra credit. There's more you can have. You could say, remove any anomalous results. This isn't an exhaustive list. You can say remove any anomalous results. There's always extra things. But I know we've whipped through that, but hopefully my little plan on the right helps. That is my top tip. You did not have to plan a practical on paper 1. It's very likely you'll have to plan a practical on paper 2. So you are all going to write down IV, DV, CV, repeats. What am I changing and how? Well, I'm changing the hand. So I'm going to make them do it with their right and the left hand. What am I measuring? How? Reaction time. So I'm going to do a ruler drop test and get them to catch a ruler. What am I controlling? Give at least 3 if you can, and then repeat it. Have a large group of people and make them all do it multiple times. Okay? Right, guys, we are done. This is recorded. You can go back. If I sped through too quickly any topics, you can go back, you can watch it later, you can quickly watch it in the morning. Joe is going to be taking over. Now, Joe's link is in the description of this video. If you are triple science, you are going to go to Joe. He is starting at 7:15. You've got a 10-minute break. The link is in my description. You've probably also been emailed it. Join Joe. He's going to be doing questions on the brain, the eye, temperature regulation, mycoprotein, I don't know, biotechnology. But for me, this is the last time I am teaching you. So can I say a massive thank you, a massive well done for paper 1. Good luck for paper 2. Top tips, read the question carefully, underline keywords, look at the number of marks. When planning your prac, IV, DV, CV repeats. And can I ask, please, tomorrow on my TikTok, I will put up a post. Please reply to me. Let me know how you found it. Let me know what came up. I do want to hear from all of you, okay? Because I've loved teaching you. I've loved teaching so many of you. It's been a blast. Let me know. Stay in touch. Good luck. I'll be thinking of you all. I will miss you. Scan the QR code if you want a last minute test. I think it's a test that you can do. And that's it. Or get some sleep. Or go to Joe. If you're triple, don't miss Joe. It's another hour and a half. Thank you. Am I going to come back? Yes, I will be back next year. If you're in year 10, you will see me again. Good luck. Please let me know. I can't wait to hear what is going to be on this paper. Please. I'm always dying to hear. So as soon as you get out, go on my TikTok and comment for me. Thank you. Oh, and I need to go because I know you're leaving. But if there's anything I have not covered that you wish I had covered, please go on my TikTok. Everything is organized into playlists. You need playlist 5, 6, and 7. Module 5, 6, and 7. Search through there. I've got videos on absolutely everything, like the carbon cycle, or what else haven't we covered? Water pollution. I don't know. Abiotic factors, biotic factors. Have a little look. Good luck. Yes, and A-level, hopefully, yeah, I do. I teach year 12 and 13, so I might see one of my Headspace course next year. Bye. Zillan, yes, hopefully see loads of you. If you do A-level biology, you need to join the year 12 course right from September, because those are the students that benefit the most, the ones that stick with me for a year and come every week and have a lesson every week, 2 lessons every week. They always do the best. So yeah, if you're doing A-level, I'll see you. I'll see you in September. Lots of hugs, lots of loves. Bye, everybody, and good luck. Bye. Bye, everyone. I'm going.